Today is Thursday, September 2nd, and we are continuing our exploration of the cardiovascular system. At the end of the last class, we started our discussion of blood vessels, and we're going to continue that discussion forward today. As we were talking before I started recording, my hope is we'll get done with the lecture part, so we'll get some time to play with the blood vessel anatomy. Like I said, I'm not going to have the opportunity to go through every single blood vessel on the list you're responsible for, but I am hopeful we can go over some of the major pathways and then also some of the trickier unusual aspects of the cardiovascular system that are worth making note of. We still have a handful of assignments due. You have 2 more unit reviews due. You have 2 more PhysioX activities, 5, which has 7 different activities to it, and a PhysioX exercise 11, where you're only doing activity 4 involving the blood. We also have another lab simulator, the Labster hematology, that is due. And remember, for full credit, you must complete the entire activity and get 80% correct. I know last time we were talking, there were some issues of people were completing the activity and it wasn't uploading properly into Canvas. Labster has been made aware of it, and it appears that the error has been fixed. So hopefully it won't continue to be a issue moving forward from that. No, that actually, it's a great question. I haven't found out about that yet. I haven't gotten an answer back on that one yet. Like I said, when I set the settings for it, it should be available to take it an unlimited number of times, but I still have not. So I sent it a notice on that. We got sidetracked by the fact that it wasn't uploading the information properly, and so I have not heard back about that one yet. But I did make that contact, and I am hoping to hear that. There might also be a way that I can reset it for individuals. So if that's an issue, we'll see about that as well. And then lastly, also don't forget, on Tuesday, your cardiovascular exercise is due. And that's kind of what we were talking about with the blood vessel anatomy, where you're tracing drops of blood going from one place to another in the body and using those blood vessels for those driving directions. And again, that's really what it kind of is. It's driving directions, as we talked about, knowing where Interstate 80 is, knowing where Madison Boulevard is, knowing where Auburn Boulevard is. All of those are useful pieces of information, but knowing how they relate to each other and knowing how you can use them to get from your house to the school is something that is much, much more important than just knowing where each individual one is. So knowing how these things relate to each other are absolutely going to be important. The cardiovascular exercise is in your modules, again, with it due on Tuesday. It's a little late to be asking that question, but again, like when all else fails, the best thing to do for homework assignments, well, there's actually really 2 ways to do this. Here, hold on, let me set this up this way. This will be even easier. So when you are in our Canvas site, when you go to the modules, under the modules, you have the assignments and stuff that are going to be due here. So notice this is where you turn in your cardiovascular exercise. And here in the lab handouts is where your cardiovascular exercise is located as well. Lab simulators are located here as well. So again, all of the activities that you're responsible for, even though the unit reviews, which obviously aren't something that are online, but it tells you exactly where to find it, right? It tells you exactly where to find it. If you were to click on the cardiovascular exercise, it reminds you that you have to fill out that handout. And so then when you go back to the module, lo and behold, as we talked about a couple days ago in class, there was this handout that you were responsible for. So we talked about the tracing the blood. You have to go to the 2 places I gave you. You need to come up with your 2. So again, we did all of this, but make sure you are completing this because, like I said, this one is graded for correctness. All right. Any other questions before we go back into the material? All right, excellent. So let's bring back up the lecture. We left off last time and we were talking about, I started the recording? Yes, I did. Okay. We were talking about the microscopic anatomy of arteries and veins and how to distinguish them from each other. And we were talking about some of the differences, like in the muscular wall of the artery, because it's dealing with higher pressure blood, the larger lumen of the veins because they're dealing with lower pressure blood. And as we kind of hinted at in the last class, arteries pretty much have most of the advantages over the veins. They are the closest to the heart, the pump, so they're dealing with higher pressure blood, and those higher pressures make it easier to set up pressure gradients that allow blood to move through them, and we'll actually see the numbers for that in a little bit. And they're also, most arteries are carrying blood with gravity down away from the heart, whereas veins are dealing with lower pressure blood. They're further away from the pump that is the heart. And again, they have that larger lumen to help with that, but they need other things to assist them as well. One of the big things that veins have to help them are valves. Now, these valves, if we look at them, you can see they're very, very similar to the semilunar valves that we saw inside of the heart. They are made up of half-moon shaped cusps, whose responsibility is to make sure that the blood only moves one way through the veins. So as blood moves back towards the heart, if it tries to go back down away from the heart, it's going to slam that cusp close and be stuck here and have no choice but to continue forward. Of course, we still need ways to help to facilitate the movement of the blood back to the heart. And there are a couple main ways we do this. And you see one example of this right here. Here we see what is known as the muscular pump. Many of our veins are a little bit more superficial. And so here we happen to see a large vein that passes right through the two bellies of a muscle that is commonly referred to as the calf muscle. Of course, we don't call it that in this class. What is the appropriate name for this muscle?
Gastrocnemius.
Excellent, the gastrocnemius. Notice, as we know, being primarily parallel muscles, when a parallel muscle contracts, not only does it get shorter, but it typically gets wider as well. So notice when we flex the gastrocnemius by doing a plantar flexion, as we see here, that squeezes the blood located within this portion of the vein. Notice because of those two one-way valves, the blood cannot go back downward away from the heart back towards the foot, and instead is forced to move through, forcing this valve open and moving closer to the heart. So we're able to use this muscular pump to be able to facilitate that movement. And there are other things that can help it as well. So these valves, just like the valves in the heart, help to facilitate and maintain a one-way movement of the blood. Now, as we mentioned, there are some distinct differences between arteries, arterioles, veins, venules, and capillaries. Here we have the pretty picture from the textbook that shows some of the numbers, but even still, even within arteries, there are two different types of arteries we want to be able to talk about as well. So while we've talked about general characteristics, let's get a little bit more specific about the specific types. As I mentioned, there are two main types of arteries. The first is an elastic artery. Elastic arteries are also sometimes referred to as the conducting arteries. With a name like elastic artery, what do you think is very distinct about them?
Elastic materials.
Yeah, they have large elastic membranes. And again, we kind of talked about why this is the case in the last class. Not surprisingly, these elastic arteries are the ones that are closest to the heart. Because as we mentioned, the heart is indeed a pump. It is not like a faucet where you turn it on and you have a continuous flow of blood, right? As we know, the ventricle contracts, and when it contracts hard enough, it ejects that blood out. And so we get a large bolus into that blood vessel. And so it needs to be able to stretch to accommodate that. And then once the heart stops pumping blood out of the ventricle, as that blood moves away, we need to maintain the pressure of it. So rather than having it stay huge and having the pressure drop dramatically, we need that elasticity to have that recoil. So these elastic arteries allow the arteries to respond to the pulsatile nature of our heart. They can expand to accommodate the bolus of blood and then retract to help to move it along its way, and then expand and contract and expand and contract. So this elasticity plays a vital role in helping in that movement and maintaining that movement of the blood. Also, being the ones closest to the heart, not surprisingly, they're the largest in diameter for our arteries. And we already explained why it has that big elastic membrane. As you move further from the heart, you end up in what we know as the muscular arteries. Muscular arteries are also known as the distributing arteries. And why do you think they're known as the distributing arteries?
Distribute out to the different limbs.
Yeah, absolutely. The limbs or the other organs that they need. Why do we need to worry about that? Isn't every single portion of our body able to get the max amount of blood that it needs all the time?
No.
No, of course not. So we need to be able to distribute the blood to the areas that need it. If you're sitting here calmly in the class, right, digesting that cheeseburger you had for breakfast, then you need blood going to your nervous system to comprehend the information that I'm saying, right? Your digestive system to break down that cheeseburger you had for breakfast, and maybe your right arm as you're doing a lot of writing. However, if you're exercising or jogging, right, then instead we need to send that blood to our legs, to our lungs, to our liver to get the glucose that we need to maintain that activity. So these blood vessels are the ones that are able to send the blood where we need it to go. And how do they do that? By changing their size, right? As a blood vessel constricts, the lumen gets smaller. And what happens to the amount of blood that goes in that direction?
It's less.
Yeah. Whereas conversely, if a blood vessel dilates, what happens to the amount of blood that is going in that direction?
More.
Increases.
Increases, absolutely. So by changing their diameter, they are able to change where the blood is going.
Professor,
That is why they have both the thickest tunica media and also why they're called distributing arteries. I'm sorry, was there a question?
yeah, just a quick question. So would the brachial artery be considered a distributing artery? Because it splits into the radial ulna, right?
Yes, so yes, and in fact, most of the named blood vessels of the body are indeed muscular arteries and distributing arteries, absolutely. So brachial is an example of a muscular artery, as is for that matter the radial and the ulnar.
Thank you.
Yep. Now, remember, as we mentioned, an arteriole is certainly defined as a small artery. And again, the book explains to us that it is the smallest types of arteries and they're typically less than 30 microns in diameter. Is that a number that's important that you need to know?
No.
No, I mean, this kind of information is good for important things like, for instance, wooing women, right? If you walk up to a woman in a bar and say, hey, did you know that arterioles are typically less than 30 microns in diameter? They just swoon when you say those kind of things to them. But again, that kind of number isn't necessarily something that is particularly important. Clearly, we know they're small. Clearly, we know, as we talked about in the last class, that their job is to directly give blood to the capillaries. But notice also, if you look at the picture, an arteriole is more than just a small artery. Notice, unlike an artery, as the picture illustrates here, it does not have all three tunics the same way that an artery does. It absolutely positively has an endothelium. You have to have that simple squamous epithelial tissue on the outside and a little bit of the areolar connective tissue off of that. But notice it has a scant or irregular, oops, I thought I said this here. It has a irregular and incomplete tunica media. Notice instead of having a big, huge, thick, continuous layer, it more has irregular bands of smooth muscle wrapped around it. Again, that smooth muscle is still going to be important for changing the diameter of our blood vessels. Certainly that can affect the flow of the blood, but this ability, again, these are tiny blood vessels, but we have massive numbers of them. So if, let's say, not so theoretically, all of your arterioles all dilated at once, what do you think would happen?
Your skin would get flush.
True. Okay. If the ones just in the skin dilated, then absolutely you would have an increased blood flow to the skin and you'd be very embarrassed. Like, for instance, if you were giving a speech in class and your pants fell down, that might happen. But what would happen if not just the ones in your skin, but all of your arterioles all dilated at once?
Blood pressure
Give
would
off a lot of heat.
Hold on. Jacob, what were you saying? I like where you were going.
blood pressure would drop dramatically.
Absolutely. You would have a massive drop in blood pressure and maybe even have difficulty getting blood to go to your brain. Luckily, there's nothing that does anything like that, is there? Well, as those of you who took me for 430 know, my daughter has a very severe peanut and tree nut allergy. So everywhere we go, I carry EpiPens with me. And why do I carry those EpiPens with me?
Okay, is it allergic reaction?
Yeah, we're worried about a large allergic
Anaphylactic.
reaction and anaphylactic shock, where you get a massive drop in blood pressure because of a massive dilation of blood vessels. Well, this is what we're talking about. And that's why you give her the epi, because that adrenaline stimulates these arterioles to contract. And when they contract, that brings the blood pressure back up. These arterioles are also known as the resistance vessels because they can play an important role in modifying, maintaining, and regulating blood pressure. So yes, arterioles are small arteries, but they're more than just that.
Would
Yeah.
like blood pressure medication, when you take it, would that affect these? Like if you
Not,
have high blood pressure and
not typically. Are there some that could affect something like this? Yes. However, with blood pressure medications, typically the two main types, one are either more diuretics to help to get more fluid out of your blood, right? Because again, when we talk about blood pressure, we're talking about the force of the blood pushing on its walls. So that's why when the walls increase, it pushes less against the walls and the pressure goes down. So most blood pressure medicines either involve some combination of being a diuretic, reducing water volume in your blood vessels. So if you bring water volume down, you bring blood volume down, you bring blood pressure down. Or some type of either a calcium blocker or something along those lines that decrease the strength of the contractions of the heart. Because as we talked about last class, if less blood is ejected from the heart into the blood vessels with less blood pushing against the walls, blood pressure goes down. So theoretically, you could probably do something like this here, but it has the potential of being so global. My guess would be it would be hard to control that in a manner that would be safe. But theoretically, probably you could do something like that, maybe. with a little more control than we probably have. All right. So arterioles, definitely small arteries, but they also are incomplete in their anatomy. They feed blood into the capillaries, and they play this important role in blood pressure. Speaking of the capillaries, capillaries are by far the smallest of our blood vessels. Basically, their diameter is the same size as a red blood cell. So our red blood cells pretty much have to line up single file to get through there. Also, notice from this basic illustration, it also has the simplest anatomy. It is just a single layer of flat cells and a little bit of a basement membrane. Essentially, it's just that one tunic, the tunica intima, or the tunica interna. And that's it. Why? Why would we want our capillaries to be made up of just a single layer of flat cells?
So they can do the exchange of materials?
Exactly. Remember, as we talked about, capillaries is where all the exchange of materials takes place. And as we know, or maybe as we don't know, but as we should know, the exchange that takes place all the exchange that takes place in the capillaries is passive. Someone remind me again what that means?
Doesn't require ATP.
Doesn't require ATP. So it's solely based on pressure gradients, osmolarity gradients, concentration gradients, things like that. And so it uses basically simple diffusion. And as we talked about in 430, one of the major things that helps to facilitate diffusion is having it go shorter distance. The shorter the distances that it has to travel, the faster diffusion will occur. So by having just a single layer of simple squamous cells, we have as basically a thin of a membrane as possible to help to facilitate that faster diffusion. Now, this is true for all capillaries, but do you think all capillaries are the same?
No.
I'm asking the question, so what's the obvious answer?
No.
No.
No. In fact, there are three main types of capillaries. Let's look at those three main types of capillaries. The first, and by far most common, is what is known as a continuous capillary. This continuous capillary is basically one we just saw the previous picture of. We have a single layer of squamous cells forming a completely intact wall to our capillary, a little bit of the basement membrane around the outside. And while certainly some of the materials that are going to diffuse into and out of our capillary can do it through the plasma membrane, just as many are going to sneak through the cracks between the cells. These cracks between the cells are what are known as the intercellular clefts. And this is an important place where fluid and solutes dissolved in that fluid are able to easily move and pass into and out of our capillaries. Like I said, this is by far the most common. So we find it in places like the skin, we find it in places like the muscle, all sorts of other places. And while it's going to let plenty of little stuff out, is that red blood cell going to be able to get into and out of this capillary?
No.
No. Large hormones or large proteins or other formed elements going to be able to get in and out?
No.
No, and that's a good thing. Just water and little stuff. In fact, in one particular area in particular, we may actually want to make these connections between the cells even tighter, helping to make this even harder to get things out. And in fact, if we were feeling particularly frisky, we might even take a particular type of specialized support cell that could wrap its plasma membrane around this, helping to form an even bigger barrier, making it even harder to get things into and out of this capillary. Where might something like that, with that really restrictive movement of materials, be useful?
Blood-brain barrier.
Exactly, in that blood-brain barrier, releasing materials into and out of the brain. And absolutely, then, someone remind me, what would the name of that neuroglial cell that would help to form that blood-brain barrier be?
Yeah.
Satellite cells?
There we go. Satellites, remember, in the peripheral, they do something similar peripheral, but in the central nervous system, it is indeed, as I heard someone say, a couple people say, actually, the astrocytes. Absolutely. So these continuous capillaries are also going to have these tight junctions, have these astrocytes, and so play a very important role in the exchange of materials in nervous tissue as well. And like I said, this is good because big proteins or large chemicals like hormones can't get into and out of this capillary. However, are there times where we might want to get large proteins or large chemicals like hormones into and out of a capillary?
Absolutely.
Yeah. So luckily, we have a second type of capillary. And that second type of capillary is what is known as a fenestrated capillary. Notice this one is almost identical to the continuous, with one big exception. Notice we have a complete layer of epithelial cells. We have those intercellular clefts in between them. But notice the big difference. The big difference here is that there are actually physical holes in the epithelial cells themselves. These physical holes, kind of like Swiss cheese, are what are known as fenestrations. And by poking holes in our cells, we make it easier for bigger things to get in and out. So this is a perfect opportunity for larger proteins or larger chemical signals to be able to get into and out of this cell. Where might something like that be useful? Where might we want to have these types of fenestrated capillaries?
Kidneys.
Kidneys is a great example. We want to be able to filter the blood there, so that's going to be really important. Anyone think of any place else where we might want to get big things into or out of the blood?
Small intestines.
Small intestine.
Right, in our digestive system, absolutely. Right, so places like our small intestine, places like our endocrine, some of our endocrine glands, our kidneys, our choroid plexus. What's the choroid plexus again? I've heard of all those other things. I'm not sure I remember what the choroid plexus is.
Isn't that part of like the amniotic sac or?
No, although that's a very good guess.
It's somewhere there.
It starts in the third ventricle, creates a cerebral spinal fluid.
Absolutely. The choroid plexus is what produces the cerebrospinal fluid and is actually found in all of our ventricles, the lateral, the third, and the fourth ventricle. All four ventricles have choroid plexus in it, and you're absolutely correct. They're responsible for making our cerebrospinal fluid. So again, these are places where we want to be able to get larger things into and out of the capillaries. But again, are really large proteins going to be able to get in and out here? And look at that red blood cell. Is that red blood cell going to be able to get in and out?
Negative.
Right. But isn't that a really good thing? Do we want our red blood cells getting out of our capillaries?
No, we don't.
Usually we don't, but I know we haven't talked about blood yet, but when you look at that red blood cell, what do you notice about it?
It's like circular in shape, or
Yeah, it kind of looks like a donut where the hole hasn't been punched through. It actually has what we call a biconcave shape, where it's concave on both sides. What else do you notice about it? What is, for instance, this cell over here, this simple squamous cell have that our red blood cell doesn't?
Nucleus.
Doesn't have a nucleus. A red blood cell is basically a big bag of hemoglobin. Doesn't have really any other organelles, doesn't have a nucleus. So can it make new proteins? Can it repair itself? Can it heal itself? No. And in fact, the red blood cell only lasts for about 100 to 120 days. So while yes, we absolutely positively want to keep that red blood cell in our blood vessels until it dies, then we need to get rid of it. And not only if red blood cells die, do we need to get rid of those old red blood cells, but then that probably means we need to get new red blood cells in to replace it. So while these fenestrated capillaries are great for getting slightly larger things into and out of our blood vessels, we need a very unique, very specialized capillary for some of these more unique jobs. Notice this sinusoid capillary is very, very different from the other two. Well, it's not entirely different. It is still comprised up of a single layer of epithelial cells. These have huge fenestrations in them. But notice also, they don't form a complete epithelial layer. It has, it has an incomplete epithelium. And this incomplete epithelium, along with those huge fenestrations, allow for the movement of large materials, big proteins, big chemical signals, and in fact, even formed elements like red blood cells to get into and out of our capillaries. So not surprisingly, if we were going to get, again, if we were going to get new red blood cells into them, where would we want sinusoid capillaries?
Liver.
True, liver is definitely one of the places that we would do it, but that wouldn't really help us get the red blood cells in. Where are we going to
Bone
get
marrow?
those? Say again?
Bone marrow.
Yeah, red bone marrow, absolutely. If we wanted to get rid of and break down the red blood cells, definitely the liver would be one of the places. Is there any other organ we learned about that can help to break down old and dead red blood cells? Yeah, spleen, absolutely. Spleen is definitely one of those, absolutely. So places like the bone marrow, places like the liver, places like the spleen, other lymphoid tissues, the endocrine organs, places where we need to get really big things into and out of the capillaries. We'll have these highly specialized, highly modified sinusoid capillaries. So those are our three types of capillaries. Questions on that? All right, once the blood passes through the capillaries, any of the three types, then as we mentioned, it needs to go into the veins. Of course, the immediate veins that receive blood from the capillaries are the, I lied. We need to talk one more thing about capillaries first. Capillaries are formed typically into structures called beds. Let's do a real simple example of this. As we know, do a real simple example of this. Capillaries are fed into by arterioles. and as I just mentioned, are fed out into venules. And then they're connected. Remember, one of the things we said is capillaries are a turnaround point. This turnaround point is sometimes referred to as the vascular shunt. That is that direct passageway from the arteriole to the venule. However, capillaries are also arranged into structures known as beds. And those beds involve multiple other blood vessels forming these big elaborate trees that come off and come together from the two sides, forming this big elaborate mesh-like structure. And these additional capillaries are what are known as the true capillaries. Or like I said, they form the capillary beds. Now, blood is always traveling from the arteriole and to the venule through that vascular shunt. But is blood always traveling through your true capillaries?
No.
Why not? Well, what's the advantage of this structure? Let's start from there. What is the advantage of having this big, elaborate, true capillary bed? What does it give us?
Multi-directional.
Pathways.
Some of it is multi-directional. I'm sorry, what was the other answer?
A lot of pathways.
A lot of pathways, excellent. And that lot of pathways gives us a large surface area. Why might a large surface area be convenient?
Distribution.
Large amount of gas exchange.
Yeah, not just
Lots
gas
of
exchange, but all exchange. Remember, exchange occurs via diffusion, and a large surface area facilitates a better diffusion. The more surface area, the more diffusion that can take place. Now, while you're sitting in your chair or laying in your bed listening to me talk, do you need to be filling all of the blood vessels in your leg, all of these true capillaries with blood, getting that massive amount of diffusion of nutrients and resources, the removal of waste, and all of those from the things going on in your legs right now?
No.
No, because they're not active. When we take our 15-minute break and you have to run 2 blocks to Starbucks to get your coffee and then run 2 blocks back, then do you need all of that blood going to the legs for all of that nutrients and all that oxygen and all of that waste removal?
Yep.
Yeah,
Yeah.
absolutely. So what these true capillaries actually have is they actually have these little bundles of smooth muscle. And these smooth muscle are what are known as the precapillary sphincters. And these precapillary sphincters can, as smooth muscle can, dilate or constrict. So as they relax, blood flows into those true capillaries. And when they constrict, they stop the flow of blood into these true capillaries. So again, I've done a truly amazing job of drawing this, but let's take a look at a pretty picture. Here we see.
And is this all based? Sorry, Professor. Is that all based off of the parasympathetic? Like that'll be a part of all that.
That is an excellent question. How do we control the smooth muscle of the precapillary sphincters?
We don't.
We don't. So your
It's automatic.
capillaries are just, well, okay, so it's automatic. You don't voluntarily control it. You can't blush on command, right? But it is controlled by something. And what is it controlled by?
Actually, that'd be fight or flight. So I guess that'd be sympathetic.
Anything else?
Hormones.
There we go. Perfect. Remember, it's our sympathetic nervous system that controls almost all of our blood vessels. There's really only one major exception. What is that one major exception? Anybody remember from 430? Okay, anybody remember and willing to say it? I'm sorry, go ahead.
Is it the sex organs?
Exactly. The erectile tissue, the penis in the male, the clitoris and labia in the females, that erectile tissue, those blood vessels are controlled by the parasympathetic nervous system. So parasympathetic nervous system that occurs during arousal causes them to dilate, causes them to be engorged with blood, where pretty much every other blood vessel in the body is controlled solely by the sympathetic nervous system. All right, back to the pretty pictures. As I mentioned, here we see, and again, we'll talk about this term anastomosis in a second, but we have this true capillary, or what is known as the vascular shunt, that goes straight through, make a color, let's see, from one side to another. Then we have the true capillaries branching off of them with those precapillary sphincters that, as we said, can be constricted or dilate to control the flow of blood. Notice, while you're sitting here resting and not moving your legs, blood is still going to the muscles of your legs. It still needs that capillary. Remember when we were talking about our driving directions, the capillary is always that turnaround point. So notice with that vascular shunt, even if an area isn't particularly active, it still needs some oxygen, still needs some nutrients, and that blood still needs to turn around and get back to the heart. But when an area is active, then we have the ability to expand the amount of blood, increase the surface area, increase the exchange of materials to meet the increased demands in that region of the body. And as we mentioned, sympathetic nervous system controls it, and chemical signals like hormones, although there's other chemicals as well. One of the ways we control blood vessels is with the use of nitric oxide. So if you're at that rave on Friday night and you happen to inhale that balloon of nitrous oxide, right, that can make your skin very flush as a result of it, because that chemical, much like the nitric oxide, dilates blood vessels and you become quite flush as a result of it. So again, chemical stimulants, drugs, hormones, all of those things, as well as primarily the sympathetic nervous system. All right. Questions on that? Now, as I mentioned, this is where the exchange takes place, and it is a passive process. So that means we need to talk about how this actually occurs. And we'll do that, but we're going to do that in a bit. We're not going to do that yet. First, we are going to follow our path back to the heart. As I mentioned, capillaries feed into venules. And unlike arterioles, venules really are only defined by two things: their size and the fact that they directly receive blood from capillaries. Even though our picture doesn't do a good job of showing it here, they really do have all three tunics, externa, media, and interna. I know the picture doesn't really show smooth muscle, although remember there's very little of it, but really they really are just small. Notice, as we also talked about, a venule typically has a larger lumen than an arteriole, because as we said, typically veins have larger lumens than arteries at the same levels, because they need that lower resistance for easier blood flow. And then again, veins are kind of distinguished by medium and large. And again, the only real difference is their size. And since numbers isn't something we really care too much about, we're not going to worry about having to distinguish the two of those. All right. Now, as I did mention, so really the only difference between venules, medium, and large veins is their size, and we don't care about those numbers. But what we do care about is their function. Notice blood isn't quite evenly distributed throughout the body. Notice, in fact, some of these numbers are a little bit surprising. This one always surprises me. I think, as I mentioned it in the last class, I read somewhere, I think every living cell in your body is something like 0.3 microns away from a capillary. So obviously we have a massive, massive, massive numbers of capillaries throughout our entire surface of our body. So how the heck, if the capillaries are that massive and everywhere, how can they only be holding 5% of the blood at any one time?
Because they're so small.
Because they're,
Yeah, exactly, because they're so tiny. Remember, they're so tiny that the red blood cells have to line up single file to get through them. Notice at any one time, only about 8% of our blood is in our heart, only about 12% in our pulmonary circulation, and the rest is systemic. But notice even there, it's not equally distributed. Our arteries, dealing with the higher pressure blood, having the advantage of gravity, is able to more easily move the blood. So typically the blood moves more quickly through the arteries. Their volume veins are much more numerous. They're much lower pressure. They have to fight gravity to get it back. And so the large portion of our blood is just slowly chugging along inside of our veins, working its way back to the heart. Now, again, as I mentioned, that's why you're sitting here calmly in class. If instead a bear with an axe walks in the room and you have to either fight that bear with an axe or outrun somebody in the room, do we have the ability to be able to constrict those veins and force the blood back to the heart faster to increase the speed of that?
Yeah.
Yeah, in fact, if you remember way back in 430, we talked about your skin being a blood reservoir. One of those areas where the blood is slowly chugging its way back. So when someone gets scared, what's one of the ways you can tell they're scared?
You get white.
Yeah, they get pale because the blood vessels have constricted to drive the blood back to the heart, back to the lungs, back to the muscles, right? Your hands get cold because they're pulling away from the surface of the skin. So you get those cold hands. Of course, your sweat glands become active, so your hands get cold and clammy. You get pale as that blood is rushing back towards your heart. So we have this systemic venules and veins that act as this reservoir, that when demand increases, we can constrict those veins and push the blood back towards the heart, speeding this rate up, which, if you remember, is one of the things that we talked about, increases cardiac output. Remember, as we said, if we could get the blood coming back to the heart faster, we could fill the heart more, and that would increase our stroke volume, and that would increase our cardiac output. See, it's almost like all this information kind of works together. Isn't that funny?
Sorry, Professor, this is like a total off the wall kind of question, but like an inversion table, like when they say, oh, put your feet up, you know, above your heart and how it helps. Like what's the benefit of doing something like that, being upside down or?
Gravity. Again, as we talked about, that's actually one of the things that I did to my wife when she was pregnant. When she'd come home from the day of work, I would hang her upside down. And why would I hang her upside down when she'd come home from work?
So you can keep track of her?
True. One of it was to keep track of her.
she's been sitting all down.
Right. Well, one of the things that happens with females during pregnancy is you get tremendous swelling of the feet and legs. Right. We'll talk about why that occurs in just a second. But raising the feet, raising the legs above the heart, give the veins then the benefit of gravity to help to drain those areas and get the blood more rapidly back to the heart. So yeah, absolutely, those kind of things can help to deal with the swollen feet, to deal with that inflammation, to help to facilitate the movement of blood. Because if you think about it, when you're in an anatomical position, most of your body is below the heart. So that means that the veins have to fight gravity to get it back to the heart. Now, luckily, they have things to help them. As we've talked about, they have a very large lumen. And again, the reason this is important is essentially friction. As you know, I think nothing of chemistry. Chemistry is stupid and unimportant, and nothing good has ever happened from chemistry. But I do really enjoy physics. And when you have two blood vessels and you have blood inside of them, the more the blood is in contact with the outer surface, the more friction you're going to produce. And that friction basically produces resistance. And resistance is the opposite of flow. So the higher the resistance you have, the less blood flows through it. So whereas here, when you think about the amount of blood that is in direct contact with the artery versus the amount of blood or vein versus the amount of blood that is not in direct contact, compare it to a larger blood vessel. Yes, if you think about it, there is indeed more blood in contact with the surface of this blood vessel, but there's also a whole lot more that isn't. All right, so basically this has less surface in contact with the blood vessel, less resistance, more flow. So that large lumen helps with the flow of the blood. We talked about those one-way valves and how that muscular pump can help in this process. But there's one other important type of pump. And that other important type of pump is what is known as the respiratory pump. All right, let's draw the ventral body cavity. Stretch out my arm so I can do a really good job of this. Perfect, excellent. Perfect representation of the ventral body cavity. And of course, as we know, there's one major anatomical structure that separates the ventral body cavity into the thoracic and the abdominal pelvic. And what is that organ? What is that structure that divides the ventral body cavity into the thoracic and the abdominal pelvic?
Diaphragm.
Diaphragm.
Diaphragm, absolutely. Now, we've talked about the diaphragm way back in 430 when we were talking about the functions of muscles, because as we know, when a curved muscle like the diaphragm contracts, what happens?
Flattens.
Yeah, all muscles get shorter when they contract, and when a curved muscle shortens, it flattens. And so as a result of that, I didn't do a very good job of lining up my diaphragm, but you get the idea. With the diaphragm there. Here, my analness isn't going to allow that to stand, so. Can I do that? I don't know if I can just change one side. Oh, I can. Oh, no, I can't. Ah, screw it. All right, I stopped caring. All right, excellent. It can, when it contracts, it flattens. As a result of that, what influence does that have on the volume of our thoracic cavity?
Increases the volume.
Increases. And when we increase the volume here, what happens to the pressure in the thoracic cavity?
Decrease.
Decreases, absolutely. As we know, our thoracic cavity is connected to the outside world via the lungs, via the trachea, via all of those fun things. And so when the pressure decreases inside of the thoracic cavity, what happens to air?
Wants to come rushing in.
Yeah, air rushes in. And we breathe in. That is how we inhale, right? We all learn that. But guess what? This diaphragm doesn't just affect the thoracic cavity, it affects the abdominal pelvic cavity as well. So when the diaphragm contracts and gets flatter, what effect does that have on the abdominal pelvic cavity?
Decreases volume, increases pressure.
Exactly. Here, our volume decreases. And as a result of that, our pressure increases. And when the pressure increases here inside of this cavity, any blood that happens to be in these vessels, or more specifically in the veins in the abdominal pelvic cavity, is that blood going to want to stay here in this higher pressure location?
No.
No.
No. So it's going to be pushed out of the abdominal pelvic cavity and try to move instead to the thoracic cavity where the pressure is less. So the same way the air wants to come to the thoracic cavity, this helps to move blood back to the heart. And let's look at the other side of this as well. As we know, that diaphragm doesn't say contracted forever. Eventually, what ends up happening?
Exhale.
Yeah, well, yeah, you're going to relax the diaphragm. And when you relax the diaphragm goes back to its original shape. And when it goes back to its original shape, what effect does that have on the volume of our thoracic cavity?
Decrease.
Excellent. And when the volume decreases, what effect does that have on the pressure?
It
Increase.
increases.
Excellent. And of course, that is going to, I'm not going to bother drawing the neck and the mouth and all of that stuff again, but you get the idea. It is going to push the air out. And as the air comes out, right, that all happens. But at the same time, this also affects our abdominal pelvic cavity. Notice now here, our volume increases. And when the volume increases here, what effect does that have on the pressure?
Decrease.
Decreases. Now, with this low pressure area, blood may be drawn to it. Can the blood from the thoracic cavity go back down in the veins down to the abdominal pelvic cavity?
If there's no valves to stop it.
But there are valves.
Yep.
So with no valves, it
No.
can't. However, can this low pressure environment help to draw blood up from the legs? Yeah, absolutely. So this low pressure environment is going to help to draw the blood pressure up, blood up from the legs. Again, you haven't thought about these terms. You may not have thought about these concepts in these terms, but I know this is something you guys are familiar with. For instance, if you have to stand on your feet working for 8 hours, would you rather do that doing something like waiting tables where you're constantly moving all over the place? Or would you rather do that standing inside of a box collecting tolls going across a bridge?
Moving around.
Moving around, right? If you're stuck standing still for 8 hours, not only are your legs going to be stiff, but your feet are going to be swollen at the end of the day. Your feet are still going to be a little swollen from standing for 8 hours for waiting tables, but at least if you're moving around, you're using that muscular pump that we talked about to help to contract those blood vessels and bring the blood back towards the heart. So that moving helps. This respiratory pump is useful until, as I just finished mentioning, you start growing a basketball inside of your abdominal pelvic cavity. As you start growing a basketball inside of the abdominal pelvic cavity, how much are those minor changes in the size of your diaphragm going to really affect the pressure in your abdominal pelvic cavity?
A lot.
A lot.
Really? All right, well, so for starters, if you have a basketball in here to begin with, so let's take that growing baby and put that growing baby here inside of your body. Is that diaphragm even going to be able to flatten out as much as it was able to before?
No.
No, and if it can't go down as much as it is before, can it change the pressure in here as much as it could before?
No.
No.
No. So do you think it's easier to use this respiratory pump with or without the baby inside of there? Without, absolutely. So that's one of the why I said plenty of healthy women, when they're pregnant, have major issues with swelling, as well as issues with breathing, right? Breathing can become much more shallow when you've got that baby growing inside. And that shallow breathing also is going to affect that ability of that respiratory pump. So it dramatically increases the problem with swelling and edema in the legs. And it doesn't just have to be a basketball, right? If you have too many cheeseburgers, 17 cheeseburgers for breakfast, lunch, and dinner, right, and start increasing the mass of adipose and other stuff inside of your abdominal pelvic cavity, can that affect your ability to effectively use this respiratory pump to help to facilitate the movement of blood back? Yeah. But that's okay, because if the blood gets stuck in the blood vessels and can't find its way back, that's not that big of a deal. If you have those valves in there, those one-way valves, but you're not moving the blood and the blood stays there super saggy and the pressure in there increases, it can't damage that blood vessel or cause that blood vessel to dilate and swell or anything like that, can it?
Yes, it can.
Yeah, and what do we call that condition?
Varicose veins.
Varicose veins. That's exactly what varicose veins are. Varicose veins are basically problems with the valves. Some of it can be genetic. Some people can be more predisposed to it than others. But typically, increased pressure in the veins can lead to damage of the valves. And that damage of the valves causes the veins to dilate. As we mentioned, the veins tend to be more superficial. And suddenly you have this big vein sticking out on the side of your leg, this spidery vein sticking out on the side of your vein because of that decrease in circulation. It's not an uncommon thing to happen during pregnancy. And the good thing is, for most women after pregnancy, they often will go away. But if the damage to the valve is severe enough, can that varicose vein become permanent?
Yes.
Yeah. Can you do anything about it?
I think there's surgery for that.
Yeah, they'll go in with a laser and a blade and destroy that blood vessel. Because after all, the most important thing in the world is that we look good. We don't worry about the health or the fact that you're decreasing your blood flow to back to your heart, limiting the movement of the blood in your veins. But as long as you look good on the outside, that's all that matters. Absolutely. So yeah, sure, you can go to get cosmetic surgery to take care of that, no problem. All righty. Questions on that?
Soapbox done.
No, that was just sarcasm. All right. Last main point. Typically, we have more veins than arteries. The advantage of this is it provides alternate routes. Again, I have to work on my analogies because normally we're all sitting together in the classroom. And I say that what if, for instance, there was major construction going on Madison Boulevard right now? Would you still be able to get here to class to be able to participate in the lecture? And of course, your answer to that would be yes, because there's lots of different routes that you can take to get to our campus. However, if you think about it, when you're trying to drive between here and San Francisco, right, aside from the horribleness that Davis is, there's also that causeway. And if there is a big accident on the causeway, how long is it taking you to get to San Francisco?
3 hours.
A really long time.
Forever, because there's no other way to get around that, right? So notice, by having alternate routes, we have lots of different options for the blood. And again, pressure is what makes the world go round. If the pressure in a vein changes 1 to 2 millimeters of mercury, that may not be a very big difference in our arteries. But in a vein, it can make a huge difference. And so if there's a little bit of congestion one way, by having alternate routes to take, it can help to facilitate, to make sure that that blood finds its way back to the heart as easily as possible. Now, we have a fancy name for these alternate routes, because of course we do, because anatomists love to name everything. And that fancy name for this elaborate network of blood vessels is what we call a vascular anastomosis. And as I mentioned, these anastomoses, the plural, IS is the singular, ES is the plural, give us those multiple routes to make sure that the blood can find its way there. And as I said, they're much more common in veins. Of course, there is a big indication in that sentence. What am I intimating at if I say they're more common in the veins?
Doesn't mean the arteries don't have them.
Yeah, it means that they can be in the arteries. Why would you want one in the arteries?
Maybe in case of a blockage or damage to an artery.
Sure, absolutely. For damage, we want to be protection, but it doesn't even have to be damage. Let's think about this. Let's say I just had absolutely positively one artery that carried blood from my upbrachial region to my antebrachial region, right through the elbow. Well, what would happen when I flexed my elbow?
Cut off blood supply.
It would kink that blood vessel. But that's okay. My hand doesn't really need any blood any of the time that my arm's flexed, right? It's not flexed all that often. So it's okay if I cut off the blood supply to my fingers every time I flex my elbow, right?
No.
No, of course not. So having them, especially in places like joints or in places where blood is very, very important to get to. Let's say, for instance, we had tissues that as mature tissues were not capable of regeneration. Would it be important to try to have multiple routes to get the blood where we need it to go to try to maintain that tissue and make sure that tissue all got the oxygen that it needed?
Yeah.
Yeah, where might two areas like that be?
Brain and heart.
Brain and the heart, absolutely, right? These, not surprisingly, we call arterial anastomoses, providing alternate pathways or collateral pathways for the blood to reach any given part of the body. In fact, and we're about to take our break, but this is a perfect opportunity to do this first. Let's talk about my all-time favorite anastomosis. Yes, I have a favorite anastomosis. Yes, I've been teaching anatomy and physiology for a long time. But remember, I am also a neuroscientist by trade. And so not surprisingly, my all-time favorite arterial anastomosis is the circle of Willis. Of course, why is it called the circle of Willis?
Bob Willis.
Bob Willis.
Good old Bob Willis. Absolutely. Bob Willis was the first individual who described that. Let's take a look at some of the general pathways of blood in our brain. There are basically, and I'll do this in dark red, three primary routes that blood gets to our brain. As you know, in your cervical vertebrae, those have those transverse foramen, and those transverse foramen help to protect, that's not what I wanted, that's what I wanted, blood vessels, and those two blood vessels coming up from the two sides are the vertebral arteries. The vertebral arteries come together to form a singular blood vessel that kind of moves up the brainstem, known as the basilar artery. And that basilar artery branches to form two big blood vessels known as the posterior cerebral arteries. And guess what the posterior cerebral arteries do?
Innervate the brain.
Yeah, and the posterior part of the cerebrum, right? A little bit of the temporal lobe, occipital lobe, stuff like that. But there is a second route that blood can take to come up to the brain. And that is to come up via the two internal carotid arteries. Those internal carotid arteries, the left and the right, just like there's a left and the right cerebral arteries, come up. And from them, they primarily branch. And when they branch, they form branches that move towards the front of the brain, helping to form what are known as the anterior cerebral arteries. Oops. Ugh. What's going on? And they have a second branch that comes off that goes to the middle part of the cerebrum, known as the middle cerebral arteries. Not surprisingly, the anterior cerebral go primarily to the frontal lobe, middle cerebral primarily to the parietal lobe. And these are the primary paths that blood is going to take to get to the different regions of our cerebrum. However, as we mentioned, our cerebral cortex, like all nervous tissue, does not regenerate and is very dependent on oxygen. So something as much as, tilting our head to the left or the right, could that slightly change the pressure in one of our internal carotid arteries?
Yeah.
Yeah, maybe a little bit. And could that slow down blood flow in one of these blood vessels? Maybe. Enough to make a difference? I don't know. But is it worth the risk? Probably not. So one of the things that we have here around the base of our brainstem are some additional blood vessels. There are two blood vessels that basically connect from the posterior cerebral to the middle and internal carotid, kind of like this, that are known as the posterior communicating arteries. And there is an additional blood vessel that actually connects the two anteriors near the front. And not surprisingly, this is known as the anterior communicating arteries. Notice what these produce is a completely circular pathway of blood. Notice, unlike all the other blood vessels that we've talked about, where blood can flow in one direction. Blood, oh, that's not what I want. Blood just flows this way, just flows this way, just flows this way, just flows this way, just flows this way. All of these arteries all carry blood in just one direction. But notice these communicating arteries are able to carry blood in either direction. And because of this, any minor fluctuations in pressure and blood flow can be compensated for by other areas. So in this fashion, all the blood going to all the parts of the cerebrum can maintain consistent pressure, maintain consistent blood flow to ensure consistent movement of blood to the regions of the brain. Now, I've done a truly amazing job of drawing this, but if we look at the pretty picture from the textbook here, we can see the exact same thing. Our two vertebral arteries coming together to form the basilar, coming off to form the two posteriors. Notice going to the temporal and the occipital lobes. Internal carotids coming to form the, oh, here it's covered by the front of the frontal, the temporal lobe there. But here we can, the middle cerebral artery, the anterior cerebral arteries. And then, as I mentioned, we have the anterior communicating, connecting those two anterior cerebrals, and the posterior communicating, connecting here as well, forming this circular anastomosis that good old Bob Willis was the first to describe. So it's known as the circle of Willis.
Professor,
Yes.
if we have, like, let's say we're following a drop of blood and then we get all the way to, like, let's say the middle cerebral artery, what would be the vein or the sinus that would take us backwards?
We'll talk about that. Like I said, I'm hoping to get to that today. Today we will get to that. We should, I'm looking at our time. We should be okay getting to that today. Absolutely.
Got you. Because everything else kind of makes sense. But when I was looking at this, I'm like, what?
The head is challenging. Absolutely. And we'll talk about why that is when we get to that. So yeah, definitely we're going to start at the top and kind of work our way down. Although I think we're starting with the heart first, but then we'll move to the head. So we definitely will talk about those things. We should get to that today. Absolutely.
Awesome. Thank you.
Okay. All right. Excellent. It would take too much. It's not a simple answer I can give you right now. So we'll work up to it and get there when we get to it in our lab today. All right, excellent. Lastly, there was one other anastomosis I referred to you as. Remember, I mentioned that vascular shunt where blood is always going from the artery to the vein. Well, technically, that is actually known as an arteriovenous anastomosis. An interconnected network that connects the artery to the vein. So that first picture named it that, and I kind of glazed over that. But now that we've had a chance to talk more about that capillary bed, we can actually see that that vascular shunt is just another example of an anastomosis, this one that actually connects arteries to veins. All right, questions on that? All right, so as I mentioned, all of this is about controlling the pressure. Pressure is what moves the blood from one location of the body to the other. And as we'll see, pressures are also what are responsible for the exchange of materials that takes place in the capillaries. So pressure makes the world go round, and we need to talk about a lot of pressure, hemostatic pressure or hemodynamic pressures, osmotic pressures, all of these types of things. But I think we need our first break first, and this is a good natural stopping point. So let's go ahead and take our first break here. We'll go back to this so it's easier to see. Our first break will be a 15-minute break. Looks like that means we'll come back at 1:32, and at 1:32 we will restart from here. Any questions before we take our first break? All right, see you in 15 minutes. All right, let's go ahead and get started. So, as we mentioned, we've kind of already talked about this. Physics is an important part of the function of the cardiovascular system. The goal is the movement of the blood through the blood vessels. And that movement of the blood is basically what we call our blood flow. And as we've hinted at, this flow is directly proportional to the differences in pressure. Again, I want to emphasize this point. If I have two blood vessels, blood vessel A here and blood vessel B, and blood vessel A has 100 millimeters of mercury at this end, and the other one has 50 millimeters of mercury. In which blood vessel is the blood going to flow faster? Is it going to go try saying that 5 times real fast. Is it going to flow faster in A, or is it going to flow faster in B?
Depends upon the lumen.
Well, again, my drawing might not be the best, but let's say that for argument's sake that these are identical blood vessels.
We don't have the difference in blood pressure.
Exactly. There you go. You're actually not able to answer this question because I haven't told you what's going on the other side. If, for instance, on the other side over here, it's 90 millimeters of mercury, and over here it is 5 millimeters of mercury, then which blood vessel has the faster flowing blood?
B.
B, absolutely, right? Because as we just finished talking about, it isn't the absolute pressure that makes a difference, that matters. It's the difference in blood pressure, the delta P, the change between the two environments that determine how fast the blood flows. And of our three types of blood vessels, the arteries, the veins, and the capillaries, which ones do you think have the steepest change, the most dramatic change in blood pressure within them?
Arteries?
Yeah,
Arteries.
absolutely. It is indeed the arteries, right? And not only that, but remember also in our arteries, we have that pulsatile nature where the pressure changes based on whether blood is being ejected from the heart or whether blood is not being ejected from the heart. And this is what it looks like. Notice at its peak, when the ventricle is ejecting blood out, it's at about 120. Notice that's systolic pressure. And again, remember, this measures the pressure during ventricular ejection. Whereas, as we mentioned, the diastolic pressure, which is about 80, is the lowest pressure when the ventricle is not ejecting blood. So early on, we can feel that pulsatile nature of the blood as it is vacillating back and forth between those two states. And again, if you've ever had a deep cut where you cut your artery, you're aware of that because you see that pulsatile nature as the blood squirts. However, if you get a superficial paper cut, do you see the pulsatile nature of the blood flow there?
No.
No.
Notice, no, because by the time the blood reaches the capillaries, there really isn't a significant difference in the blood pressure, whether the heart is ejecting the blood or not. And notice also this point, the blood pressure when it enters the capillaries is about 35 millimeters of mercury. So if you think about it, the change in our arteries is from as high as 120 down to 35. Whereas by the time it enters the capillary, it's at 35. Notice by the time it leaves, it's at about 17 millimeters of mercury. And of course, when it leaves the capillaries, it enters into the veins. And if I cheat and put us on the other side, what does it look like the pressure of the blood is when it actually reaches back to the right atrium?
0.
Exactly. The pressure of the blood when it reaches back to the right atrium should be 0 millimeters of mercury. So notice the change in the arteries is from 120 to 35. How big of a change is that?
85?
85, excellent. Through the capillaries, it's 35 to 17. How big of a change is that?
18.
18. And in all the veins, from the tip of the capillaries at the tip of your toe all the way back to the heart, how big is the change in pressure in the veins?
17.
17. So remember that flow, that difference in pressure is determined by flow. Veins have the smallest pressure gradient in all of them. And so not surprisingly, as we mentioned, blood moves slowest through those veins. Moves fastest through the arteries, because even when the blood isn't being pumped out of the heart with a low pressure of 80 millimeters of mercury, the difference between 80 and 35 is still pretty huge. So it's these changes in pressure that allow us to determine how easily the blood is going to flow. Now, as we mentioned, our flow is inversely proportional to the me part is so let me rephrase that. Flow is directly proportional to our difference in pressure. So if we were to write this out as a physics thing, we would say that flow is equal to delta P. So for instance, as we talked about with that blood vessel before, if the pressure gradient from one side of it to the other was 50 millimeters of mercury, and suddenly it became 100 millimeters of mercury, what would happen to our flow rate? If the change in
slow down.
pressures doubled, what would happen to our flow?
Double.
It would double as well, exactly. These are directly proportional to each other. Let me write this bigger so it's prettier. Flow is directly proportional to delta P. But flow is also inversely proportional to the resistance. And as we talked about, the resistance is the amount of friction that that blood has to overcome as it is moving through. So if, as we were talking about before, I was to suddenly double the resistance by narrowing the blood vessel, what would happen to my flow rate?
Decrease by half.
It decreased by half, exactly. So are knowing the delta pressures, are knowing the resistance useful pieces of information?
Yes.
Absolutely. However, knowing what causes that resistance is even more important. And there are three main factors that can influence the resistance of our blood vessels. Oops, why are those all coming up at once? All right, well, I don't care. The first is the blood viscosity. What do I mean by that?
Like the
How thick it
thickness
is.
of the actual blood.
Yeah, I like that, the thickness of the actual blood. So if you were to increase, or let's take, if you were to increase the water in your blood, what would happen to the viscosity?
Decrease.
Decrease.
Right, viscosity would decrease and it would flow more rapidly. When you start becoming really dehydrated and water levels in your blood go down, your blood becomes thicker, it becomes more viscous, and it slows down as a result of that. Now, obviously, that can be a bad thing. When you become very dehydrated, right, that can cause major problems, not just in blood viscosity, but in other issues as well. However, is that something that happens to you instantaneously?
No.
Nah.
No, if I wanted to cause dynamic changes in my blood pressure, would this be a good way of doing it?
No.
No.
No, absolutely not. Now, again, in extreme cases, something like this can happen. If I donate a pint of blood, do I have to be careful about how quickly I get up afterwards, or what how vigorous of my activities are immediately after giving a pint of blood to helping people out?
Absolutely.
Yeah, definitely.
Yeah, because by reducing the blood volume, I reduce the blood viscosity as a result of that, and pressure can drop as a result of it. And at the same time, if instead of being magnanimous by taking out that blood to give to the needy, let's say instead I store it in a refrigerator because I have the Tour de France coming up next week. And so after a couple weeks of growing back those red blood cells, the night before the race, I take that pint of blood and stick it back into my arm. Right? The nice thing about that is now I have a massive number of red blood cells that are going to increase the oxygen carrying capacity of my blood, and I'll be able to pedal my little bike even further and faster without getting tired. But is that going to dramatically affect my blood viscosity?
Yeah.
Yeah, absolutely.
Isn't that what blood doping is?
It's exactly what blood doping is, right? It's horrible for your heart, can do severe damage to you long term, but as long as you win the race, that's all that matters, right?
Absolutely.
Excellent.
So there's actually no doping in blood doping. It's just blood. Sorry,
For
that was a
what?
question.
No, it's a great question. Yes, blood doping is basically originally was taking that blood and shooting it into your arm. There are more sophisticated ways that they can do that now. For instance, there are hormones in our body that we'll actually talk about next week that can cause you to produce more red blood cells. So if I were to take that hormone to increase the number of red blood cells that my body was making, would that increase the oxygen carrying capacity without actually having to pour a pint of blood back into my body? Yeah, absolutely. But it would still have the same effective viscosity, and it's equally as cheating, which is why they take blood. When they take blood, they're looking for the preservatives to make sure that you didn't store that pint of blood and pump it into your arms. They're looking at those hormone levels to make sure that you don't have inappropriate amounts of those, and all those things that, Lance Armstrong did. All right, questions on that? So again, in extreme cases like that, can you get an immediate effect if you pump a pint of blood into your body? Is it going to instantly affect your blood viscosity? Yes, but that isn't something that hopefully we do on a normal basis. Blood vessel length as well. I read somewhere, and again, I don't remember even if these numbers are correct. It's not important, but for every 10 pounds of weight you gain, you add an additional, mile of blood or vessels, or I don't know, some random number like that. It doesn't matter. But as you grow, get taller, gain weight, as you increase and change the number of blood vessels, and the blood vessels get longer, the longer something has to travel through it is the more resistance they're going to be. Yeah, if you're drinking your shake, it's nice to have that nice big large lumen McDonald's straw. But if you put 10 of those straws together and tried to suck a shake from across the room, would that be something that would be easy to do?
No.
No, because the further that shake has to travel in the straws, the more resistance. So gaining and losing weight can affect the number of total blood vessels. But is it super easy to gain or lose 10 pounds in a single day in 30 minutes?
Unfortunately, no.
No, exactly. So again, these are things that can have long-term implications on resistance, but they aren't something that we can use to dynamically change it moment to moment. But do we even need to be able to change our resistance and our blood flow from moment to moment?
Yeah.
Yeah, absolutely. And luckily, we have a way to do that. That primary way from moment to moment that we can influence the resistance and therefore influence the flow of blood is by changing the diameter of a blood vessel. Here is a math equation that you don't need to be able to worry about. But I am going to throw a number out there just so you can see. Right? This resistance is related to 1 over the radius of a blood vessel to the 4th power. So let's say, for argument's sake, you doubled the radius of your blood vessel, right? So you dilated and doubled it. What effect would that have on the resistance?
It would get smaller.
How much?
16, 1 over.
Yeah, 1/16, exactly. It would be 1/16. Now, again, this is not an equation I'm going to make you do on the exam or anything, but the point I'm trying to emphasize, and let's do it the other way. If I halved the diameter of the blood vessel. If I constricted it, then how big of a change in resistance would that be? Not a trick question. We just flipped it over.
2.
The resistance would be 16 times greater. Through this equation up here, I made you do some quick math to see this, not because you need to memorize this number, but the point I'm trying to make is that if you notice, even small changes in diameter of a blood vessel, can that have a huge effect on the resistance and therefore a huge effect of the flow on our blood?
Yeah.
Yeah, absolutely. So again, you don't have to memorize this equation, but just appreciate that this minor changes in this blood vessel diameter can have a huge impact on the resistance and therefore a huge impact on blood flow. Now, as we also talked about, blood pressure is basically the force of the blood on its walls. It's basically the blood molecules that are in here pushing on its boundaries. Kind of similar to what we talked about with gases. If I put you in a room, all of you in a room, and blindfolded you and let you wander around, every once in a while, one of you would bounce into the walls. And every time you bounce into the walls, I would count it. And at the end of the minute, however many times you bumped into the walls, that would be our pressure in that environment. If I then, instead of in our classroom, threw you guys all in a closet together, what would happen to the number of times you bounced into the walls there?
Increase.
Yeah, it increased. Right? So it's the same thing with blood. Blood bounces off the wall of its vessels, and that bouncing off the wall of its vessels determines our blood pressure. Most of you at this point in your life have had your blood pressure taken. I mentioned I have mine taken every 3 days, right? And again, I took it just a couple days ago. And as I said, it's a good solid 417/225, right where I like it. And when we do that, typically it's taken in either the large elastic blood vessels that are near the heart, or the muscular ones like the brachial that we talked about, bigger blood vessels where we can still see that pulsatile nature. And even though I've already given it to you, when we talked about that 120/80, what is the unit that we use with that? Even though I've said it like 10 times already so far.
Millimeters of mercury.
Millimeters of mercury. What does that actually mean? Anyone know? When we talk about pressures in terms of millimeters of mercury, what does that actually mean? Does anybody know?
Does that have something to do with like the pressure to raise a certain amount of mercury in the,
Absolutely.
when you measure the
Mercury,
pump?
mercury is a metal that at room temperature is a liquid. And so basically it is the force that has to be exerted on a bubble of mercury to get it to move a certain distance. Absolutely. So it really literally has to do with the movement of pressure, the movement of a liquid, in this case, a liquid metal. Absolutely. And so as we said, those differences in the blood pressure from one environment of the artery or vein to the other is what moves the blood. And we saw that here, right? Higher pressure, the blood moves away from it. Moves faster in the arteries, slowest in the veins. Now, as we mentioned, there is a pulsatile nature to this. So we need to know the mean arterial pressure. Now, luckily, we know that the high is 120. The low is 80. So what is the mean? What's the average going to be? Come on, you shouldn't even have to take off your shoes and socks to be able to answer this one.
100.
100 millimeters of mercury. Perfect. And so sure enough, when we look at our chart, we see that the mean arterial pressure is right here at 100. Excellent. Questions on that?
I mean, 90.
Wait, oh, you're right. It isn't there. Well, why the heck not? Isn't that how you figure out an average? You take the high, you take the low, and you figure out what's the difference in between them? Just like on the exam.
Usually.
Just like on the exams. When Amanda gets 100 and everybody else gets 10 in the class, then the average between those is 55, and that's the average for the class, right?
No, because more people got 10. You have to do it by the amount of people in the class, not just by the 2 score.
Bingo. Remember, the systolic pressure is the amount of time that the heart is actually ejecting blood out of the ventricle. Does it eject blood out of the ventricle for the exact same amount of time that it is not ejecting blood out of the ventricle?
No.
No. And since those two times aren't equal, we can't just take an average to figure out what the average arterial pressure is. Luckily, there is an equation we can use to determine this. All you need to know is your cardiac output, and you multiply that times the total resistance of all of the blood vessels in your body. Now, how do you calculate cardiac output again?
Heart rate, stroke volume.
Excellent. How many people could determine their heart rate right now if they wanted to? I'll give you a minute. How many people here can figure out their heart rate? You don't actually have to do it, but just tell me if you could do it if I give you a minute.
Yeah.
Yeah.
Yeah,
Yes.
absolutely. Excellent. In that same minute, how many of you could figure out your stroke volume? Anyone?
No.
Probably
No,
not
I
too
don't think so.
easily. And how many of you just off the top of your head happen to know the total resistance of all the blood vessels of your body?
Nope.
I calculated last
No.
night.
Calculated it last night. You had a little bit of free time. Absolutely. Yeah. Clearly, this is useless to us. Luckily for us, we have a better way to figure out the mean arterial pressure. And that is to take our diastolic pressure. And for our typical person here, what is our diastolic pressure?
80.
80. Excellent. And from that, we add 1/3 of our pulse pressure. Well, now we have one more issue. What the heck is the pulse pressure?
Systolic pressure.
It's not the systolic, because then we would just say systolic, right? I know anatomists are jerks, but they're not quite that big of a jerk. What is a pulse pressure? Anybody know?
I think because it goes all the way to the heart.
Not a bad guess. The pulse pressure, though, is actually the difference between the systolic pressure and the diastolic. So in this case, what would the pulse pressure of our typical completely unrealistic person be?
20.
Well, it would be our systolic pressure minus our diastolic pressure. What's our systolic pressure?
120.
It's 100. Sorry.
20. What's our diastolic?
80.
So what is our pulse pressure?
40.
There we go. 40. So you take your diastolic, to that you add 1/3 of your pulse pressure, which is 40. What is 1/3 of our pulse pressure?
13.3.
Yeah, about 13. So you add that together, and lo and behold, your mean arterial pressure is 93 millimeters of mercury. Obviously, this pulse pressure is important for helping us to determine our mean arterial pressure. And this is an equation you absolutely positively need to know how to do. And therefore also this one here. But also from a clinical standpoint, sometimes, especially in elderly individuals, pulse pressure is actually more important than the actual systolic and diastolic. The difference between the two can be much more of an important indicator of health in some instances than the actual individual numbers themselves. All righty, so there you go. We now know how to calculate our mean arterial pressure. We know how to calculate our pulse pressure. Of course, to do all of this, we actually need to know our systolic and diastolic pressure. And for that, we need to know how to measure a blood pressure. How many people here have actually taken a blood pressure before? Ervik, yes, you had a question?
Yes, Professor, I actually have a question about the previous chart, the graph actually.
Mhm.
So if we have like billions of capillaries and I don't know, about 100 of veins, I don't know the number. And as I imagine the surface area from for the blood flow, it decreases, right? When it goes from capillaries to the veins.
Mhm.
So how is that the pressure is the lowest in the veins and not in capillaries?
Because the capillaries, because the size of the capillaries themselves are very, very small, right? And so they tend to have a higher pressure that is still going to put them into a lower pressure vein. Again, for me, I always think of this in terms of traffic flow. And for me, I always think of it, if you're going to Davis, as you're coming down 80, there's that point where 80 before the merge basically goes down to one lane. And then that one lane merges into 80 that's going across the causeway. And there's about 6 lanes there. So no matter what type of day you're driving towards Davis, when you hit that merge, there's always a bit of a backup because you're dealing in a very small capillary that is feeding blood into a much larger vessel.
Yeah,
So
for
because
some
of the
reason,
large, so it's
we
still,
have-
you've got hundreds of capillaries feeding into just, one venule, but there's still the relative size of the capillaries versus the venules. The venule is still huge in relation. So even though you have a lot of blood pouring into it, it has a lot more space.
So if the pressure in the vein, vena cava, for example, is 0, how is it even entering the right atrium?
It's 0 by the time it reaches the right atrium. So right before that, it may be 0.5. A little before that, it's going to be 1. A little before that, it's 1.7. So when notice it doesn't really reach 0 until it pretty much gets back to the atrium. And again, remember, we have things like the respiratory pump that is helping to push it along its way.
Okay. Yeah, for some reason, I thought it decreases and then increases.
Nope.
Like the lowest is in capillaries for the nutrient exchange or something.
Lowest volume, yes. Like I said, the capillaries are so tiny, they're one blood vessel in diameter. So yes, they start big blood vessels, get to a tiny blood vessel, and bend them back up to big blood vessels. So size changes, but pressure constantly decreases.
Okay, thank you.
Yep. Any other questions? All right, so notice to calculate all of these things like the pulse pressure and the mean arterial pressure, we need to know our blood pressures. How many people here have taken a blood pressure before?
Yes, me.
Excellent.
Yeah.
What is the device you use to take a blood pressure with?
Sphygmometer. However you.
Close. You got the right idea. Absolutely. It is a sphygmomanometer. And again, that's not exactly how it is pronounced, but that is how I pronounce it because that's how you spell it. And remember, spelling does matter. You can almost be guaranteed at some point on the exam, you're going to have to spell that word. Sphygmomanometer. And you use that along with a stethoscope to listen for Cardiovascular sounds. What does that Cardiovascular sound like?
A pulse.
Okay, true. Can you be a little more descriptive?
The love thing, kind of.
Okay. Is it more of a metallic sound? Is it more, what does it sound like?
Oh,
It's kind of like a whooshing sound, right? That whooshing sound. Someone explain to me how taking a blood pressure works. Anybody know?
Yeah, you put the cuff and then you put the stethoscope and then you squeeze and then you let it go and you just see and you hear. The first one, it goes for the SP and the second one goes for the DP.
All right. Excellent. I like that. That's pretty good. Absolutely. So we get a blood vessel. Like, for instance, we were talking about our brachial artery, and you put a cuff around it. And as we've talked about, this blood vessel is close enough to the heart where its pressure is pulsatile. Where again, we'll use a fake person where it's 120 systolic and an 80 diastolic. We then take this air cuff and put this air cuff around the arm and continue to inflate it until the pressure becomes what?
180.
All right, 180, maybe 200. Right, if you really don't like the person, you take it up to 275. Right, you inflate that to about 180. Why do you inflate it to about 180?
Because you need to let out some of the air to.
Well, what
unclog
happens?
the artery.
What happens when you get that pressure to 180?
It good.
Closes off the artery.
Yeah.
Exactly. At 180, notice the pressure is now greater than in the blood vessel. And so as a result of that, the blood vessel collapses. We squeeze the blood vessel tight.
Not
And then
your blood vessel.
I'm
Because you're at
sorry?
4 something. Not your blood vessel. You're at 4
Yeah,
something.
no, they have to take mine up to like 560 before they finally let it go. Absolutely. It's like a big, huge balloon on my arm. But above, and it collapses the blood vessel. And then you're right. They slowly start taking it down. 175. Whoops. 175. 170. Right. 150. 140. 130. Right. And then finally it gets down to like 118. At 118, or really at any pressure less than 120, what happens at this point?
The needle pulses up.
True, although technically if you watch, the needle will actually start to pulse before that as the blood pushes against that collapsed blood vessel. So can you just watch the needle to know what the blood vessel is, what the blood pressure is?
Yeah.
No, you can't, because it will start to deflect before you actually can hear it. That's why you need the stethoscope, because what happens is the blood pushes against before it can open up the blood vessel, it pushes against the cuff, and so you can see. But here's the key: once it drops below that 120, now when blood is being injected, the pressure is greater in the blood vessel. And when it's greater in the blood vessel, it forces the blood vessel open. And a little bit of blood enters the blood vessel. Oops, what happened here? Now, does this last for a very long time?
No.
No. And is the little bit of blood that's able to get in there, is it going to move nice and smoothly through this blood vessel?
No.
No. So what happens is that little bit of blood produces turbulence. And as it produces that turbulence, you hear it as that whooshing sound, that Kardikov sound. And that continues as you continue to drop. As you continue to drop the pressure, 110, 100, right, 95, 85, and so on and so forth. Every time the blood is being ejected by the heart, you hear that little whooshing sound. Until the pressure in the cuff becomes less than 80 millimeters of mercury. What happens then?
You stop getting turbulence.
Right. What happens now is now you're no longer able to collapse the blood vessel. Even at its lowest pressure, the pressure in the blood vessel is now greater than the cuff. And so the blood vessel stays open all the time. And with the blood vessel open all the time, the blood flows smoothly. And when it flows smoothly, no more turbulence, no more sound. And so as someone mentioned earlier, the very first whooshing sound you hear tells you what the systolic pressure is. The very last whooshing sound you hear tells you the diastolic pressure. And so by producing this turbulence in the blood vessel, we are able to measure those pressures. And again, if you don't like my picture, we got the pretty picture from the textbook that does a nice job of doing this, showing this, and explaining. Your book does a great job of explaining this process as well. All right. Now, we could probably spend close to the rest of this semester talking about different blood pressures and the meaning of them and how they change from age and all the things that they tell and the different conditions. And so I'm painting with tremendously broad strokes. But in general, as we've talked about, you're looking for a systolic within 100 and 140 millimeters of mercury and a diastolic between 60 and 80. Anyone who has a systolic below 100 is often considered to be hypotensive. But hypotension can actually become an acute condition. If, for instance, you've been laying down for an hour and a half and you jump out of bed super quickly, your heart has been beating rather calmly in your chest while you were laying down, because your body being horizontal, it's not hard to get the blood where it needs to go. And if you suddenly stand up instantly, there's a brief moment of time where your heart has to begin to compensate to be able to force the blood to your head. And so can you get a little bit woozy, a little bit dizzy from standing up too quickly?
Yeah.
Yeah, absolutely. And that can be caused by as little of a drop in 20 millimeters of mercury can cause that type of dizziness. And then typically anybody above 140, above 90 is considered to be hypertensive. Now, again, I know there's pre-hypertensive and there's more, like I said, big broad strokes. But again, I just wanted to talk in general terms about this here. We're not going to spend a tremendous amount of time talking about blood pressure and hyper and hypotension too much.
Professor?
Yes.
What if the systolic and diastolic numbers are quite far apart from each other?
Well, it depends on the condition. Again, that's why pulse pressure can be important. Also, the systolic and the diastolic aren't equally important from a diagnostic standpoint. From a diagnostic standpoint, and again, please don't call me out on this. Like I said, I'm painting with incredibly broad strokes here. But in general, which of the two are kind of the more important number? Systolic or diastolic?
Systolic.
Systolic? Which one's typically more dynamic? Which one changes more?
Systolic.
Systolic. So that means the diastolic is actually slightly more important. The systolic is much more dynamic and a lot of things can change it. One of the things that you commonly see is white coat syndrome. When people go in to get their blood pressure changed, just the stress or concern about having a high blood pressure, can that cause their blood pressure to rise as a result of that?
Yeah.
Yeah, definitely.
You can see as much as a 30 millimeters of mercury increase in someone's systolic just from the stress of trying to take their blood pressure. Now, is that stress of trying to take a blood pressure going to cause a 30 millimeter change in the diastolic?
No.
No, probably not. So that can be. So big, huge differences between those numbers can mean numerous things. And again, it also, these, again, so broad strokes, this is almost meaningless. Like I said, as we age, these numbers change dramatically. And so to answer, one of the ways we can answer your question is it depends a lot on the age. One of the things that typically happens as we age is that we get much more dramatic change between our systolic and our diastolic. And the reason for that is our blood vessels start to lose their elasticity. If you take a large amount of water and force it into a jar, if you take a garden hose and put the spray on and try to force it into that jar, what's going to happen?
It'll spill out.
Yeah, it bounces off that wall and doesn't just spill out, it sprays out all over the place, right? Because there's no elasticity to it. And that's what can happen. As our arteries start to become less elastic, as you're forcing the blood into it, they stretch less. And if they stretch less, you can see a dramatic increase in pressure. And if they're not stretching as much, that means they're also not going to recoil as much. So as soon as the blood goes into it and starts to go away, there's no recoil in the blood vessel. And so the blood pressure drops dramatically. So it is not uncommon to see a larger pulse pressure in older people just because of the loss of elasticity of their arteries. Now, can that loss of elasticity be an indication of arterial sclerosis or other types of medical conditions? Yeah, absolutely. So like I said, I wasn't kidding when I was saying literally we could probably spend the rest of the semester talking about blood pressure. There's, like I said, there's so many implications, age and physical conditions and all these other factors, stress and all these other things that can affect it. So like I said, we're painting with very, very broad strokes with these numbers here. But no, definitely there's a lot more to it. I'm sorry, go ahead.
I do agree with what you said, because I do work as a nursing assistant and I see a lot of dialysis people do have a lower blood pressure, but other than old people, they do sometimes have more than 140 and up.
Yeah, and again, yeah, genetics can be a factor. People can be predisposed to arteriosclerosis, to less elastic blood vessels, to higher blood pressure or lower blood pressure. Addison's disease can cause hypotension, other type of endocrine disorders. Yeah, like I said, we could spend the rest of the semester talking about blood pressure. So, which is interesting, but not the point of this class. All right, so I appreciate big, broad strokes there. But again, the point is that in being able to measure this blood pressure, we're able to do things like measure the pulse pressure, figure out the systolic and diastolic, figure out the mean arterial pressure. And it is something we want to control. So we will obviously talk about the regulatory factors and the ways that we control that. However, this is as far as I want to go today. I want to make sure we have a good amount of time because you've got that cardiovascular exercise due on Tuesday to spend a little bit of time talking about the blood vessel anatomy. So I think this is a good stopping point for our lecture. So let's go ahead and stop the lecture here. We'll go ahead and take our final break at this point. And it's 2:19 now. So that looks like that's going to be what, 2:34. So we will come back and restart at 2:34. And I will start the recording at that point.
I will do have a question. Is it if I ask it in a break time?
Sure. It's not a question about the material we were just
Not about this one. No, not.
Okay, then yeah, absolutely. We can ask that. You can ask that during the break. So let's go ahead and take our 15-minute break. I'll see you guys at 2:34. All righty, let's go ahead and get started. Any more questions before we get started? Juan had a great one before the break, so I was just curious if anybody else had any other questions about that or anything else before we dive into new material. All right, excellent. Stunned silences. You know how much I love those. All right, excellent. So one of the points I have been trying to emphasize, and hopefully you've been aware of while you've been doing your driving directions, is when we talk about the aorta, I have been encouraging you, and dare I say requiring you, to divide it into distinct regions. So when you're giving driving directions, you don't just say the aorta. How many different regions of the aorta are we actually responsible for?
3.
3.
Close. Well,
Four.
let's list them and see if we can figure it out. What are
Four,
they?
yeah.
What's the first one?
Ascending aorta.
And that leads into the
Aortic arch.
which feeds into the,
Descending aorta.
although we can be more specific than descending.
The abdominal.
There we go. Thoracic aorta, which then feeds into the
Abdominal aorta.
Yeah. Now, you guys are correct. Some books, sometimes some people will lump the thoracic and abdominal aorta together into what is just simply called the descending. So I fully appreciate where some of you were saying 3 from, but I think that defeats the purpose, right? The whole point of this isn't to make this class more challenging for you. It's in fact the opposite. Whoops. Well, erasing that makes it harder. Excellent. The reason I like to divide the aorta up into these 4 distinct regions is because these 4 portions of the aorta are responsible for sending blood to specific parts of the body. And so when it comes to driving directions, understanding what comes off of where is very, very important. Let's start easy, especially because I think we've already talked about this. How many blood vessels actually, how many main primary blood vessels come off of the ascending aorta?
2.
2. Absolutely. Anybody remember what they are?
The brachiocephalic trunk, and then the common carotid artery.
Nope. For the ascending aorta.
Oh, the coronary arteries.
Absolutely. All right. So when when that oxygen-rich blood leaves the left ventricle, it exits the aortic semilunar valve into the ascending aorta. And there are only 2 branches that come off of the ascending aorta: the right coronary artery and the left coronary artery. And where do they deliver blood to?
Yeah, within the heart.
Yeah, to the heart itself. The walls of the heart, right? In fact, they start what we referred to before as the coronary circulation. Now, the coronary circulation has a lot of anastomoses and can be a little challenging. So let's take a look at it. Notice, first thing that should step out is there is some asymmetries, which isn't surprising because we know the heart is asymmetrical. We know, for instance, the left ventricle is thicker and larger and circular, and the right ventricle sits on top of it. But what should stand out right away is the big difference in the lengths of the right and the left coronary arteries. The left coronary artery is very short. It's actually very shy. It actually is hidden behind what's been erased here, which is our pulmonary trunk. So the pulmonary trunk here normally hides it. It's almost completely behind the pulmonary trunk. So notice, to get a good view of it, they had to move that pulmonary trunk out of the way. And pretty much it comes to the end of the pulmonary trunk, at which point it branches. And like most systemic blood vessels, when a blood vessel branches, it typically takes on a new name. One branch of this goes down into this indentation between the ventricles of the heart. What do we call the indentation that is there on the anterior surface of the heart between the two ventricles?
Interventricular septum.
And you get partial credit for that on the exam.
The coronary sinus?
No, what did he forget?
Left.
Well, not so much left, but you got the right idea. Is there just one interventricular septum?
Oh, there's one on the back too, yeah.
Yeah, there's one on the back too. So how do we distinguish the one on the front? There you go. Someone's got it.
Anterior.
The anterior interventricular septum. Or sulcus, sorry. Sulcus, the indentation. Now, conveniently for us, there happens to be an artery that sits in the anterior interventricular sulcus. And what is that artery?
The anterior
artery.
For once, anatomists don't hate us on that one.
Yeah, exactly, right? They make it super easy. And it's the same thing with the vein as well, right? The vein that, because again, they often run parallel to each other, is the anterior ventricular vein, correct?
I take back what I said. That's probably not it.
Exactly. It should be the anterior interventricular vein, but as you mentioned, absolutely, your anatomists hate you. So what did the anatomists call it instead?
The great cardiac vein.
There you go. Excellent. So
Why did they call it that? Like, why would they?
it was, they just explained it to you. Anatomists hate you. They're horrible, horrible people. I honestly, I can't think of a better reason. I mean, you're right. You call it the anterior interventricular sulcus, you call it the anterior interventricular artery. Why would you not call it the anterior interventricular vein? Now, it's called the great cardiac vein because it is the largest or the longest of the veins, but I don't know. Anatomists are horrible people. That's why. That's how you end up with a ciliated pseudostratified columnar epithelial tissue. It's just because they're horrible, horrible people who don't like it. All right, excellent. But notice
the second language would be the easiest one to remember.
I'm sorry?
For me, as a second language would be the easiest ones to remember.
Well, good. Well, so if the weird ones would make it easier to remember, then that's even better. Now, occasionally they give us something that is worthwhile. And again, like I said, anterior interventricular artery is not bad, as is the other blood vessel that branches off of the very short left coronary artery, and that is the circumflex. Circumflex kind of sounds a lot like circumference, and that's actually where it gets its name, because on the left side here, it wraps around the circumference of the heart, basically in the indentation between the left atrium and the left ventricle. And what do you think we might call that indentation between the atrium and the ventricle? Yeah, sulcus, the atrioventricular sulcus. Excellent. So in that atrioventricular sulcus, that indentation, we have that circumflex artery. Notice that's very, very different from what's happening over here with the right coronary artery. The right coronary artery is much longer. In fact, not only does it, I don't want to use blue, that's going to be confusing. Not only does it go around kind of the circumference all the way onto the back side of the heart, notice it also is going through that indentation between the atria and the ventricle, that atrioventricular sulcus on the right side. So it's clearly much, much longer if it's going all the way around onto the posterior side of the heart. But you will notice that on the lateral aspect of the heart is a blood vessel, kind of a small blood vessel, that travels along the edge, or dare I say, the margin of the edge of the heart. And what would we call that blood vessel that travels on the edge of the heart?
Marginal artery.
It's the marginal artery. So of course, the artery sitting next to it is the marginal vein.
Of course.
Right? No, in fact, it is the small cardiac vein. But I've got a trick to help you with that as well. If you marginalize someone, do you make them feel good about themselves?
No.
No.
If you marginalize someone, you make them feel very small. So the small cardiac vein basically runs next to the marginal artery along the lateral edge of the heart. All right. Let's flip this over and see what's happening on the backside. Notice as we follow that on the backside, that right coronary artery that we left off on, that we mentioned goes to the back, doesn't just come all the way around to the back, but notice it goes all the way to that indentation on the backside of the heart. And what would we call such an indentation?
Posterior intraventricular artery.
The posterior interventricular sulcus. And when it branches to go down there, what does the right coronary artery become?
The posterior intraventricular artery.
Which of course is also the name of the vein, right?
No.
Negative. But
Middle, cardiac.
exactly, we've had great, we've had small, so that means the vein back here is the middle. Cardiac vein. So again, they screw us up with the veins, but everything else pretty much makes sense. Lastly, notice that our circumflex artery comes around that atrioventricular septum and forms a second marginal artery. Notice this second marginal artery isn't on the edge the same way it was over on the other side. Notice here it's over here on the edge. Instead, this posterior marginal artery is actually primarily associated with the left ventricle. And why would the left ventricle maybe need another blood vessel associated with it?
The amount of muscle?
Yeah, it has a much thicker muscle. So that thicker muscle needs a little bit more juice. And so we have a marginal artery, or what is sometimes referred to as a marginal artery of the left ventricle. And notice there is a vein that empties that left ventricle as well. And it is known, because it's on the backside, as the posterior cardiac vein, and sometimes called the posterior cardiac vein of the left ventricle. So notice there's this extra blood vessel, artery, and vein associated with the left ventricle because it has a thicker muscle wall, needs more blood. And the last thing that I want to point out is notice while those veins are a little wonky in their names, we have that great cardiac vein that is the biggest one because it comes all the way from the front to the back. Small cardiac vein coming from the right side, posterior cardiac vein, and middle cardiac vein, all feeding into this big, loose, pouch-like blood vessel that collects all of the blood. And what do we call this big pouch-like blood vessel that collects all the blood from the heart?
Coronary sinus.
Exactly. And that's basically what sinus is. A sinus basically means a big, loose vein. And as we talked about, having a large lumen, very loose vein is going to help to maintain a very consistent flow, make it very easy to collect the blood. And notice, as we talked about, that coronary sinus is the third blood vessel that feeds blood into the right atrium. We have the superior vena cava, we have the inferior vena cava, and the coronary sinus. Those are the three blood vessels that feed blood into the right atrium. Questions on the heart? Excellent. Let's cheat. Go away from the pretty pictures and draw some of our own. From the ascending aorta, if you don't enter into one of the two coronary arteries, where does blood go?
Aortic arch.
Aortic arch. Excellent. How many blood vessels come off of the aortic arch?
3.
Excellent. What are they? Excellent. So the first one, I like that, is the brachiocephalic trunk. So, couple things in a name. We just identified what a sulcus is. Pardon me, what a sinus is. A sinus is a big, loose vein. We need to identify what a trunk is. A trunk is typically a large, short artery that branches almost immediately. And this brachiocephalic trunk, as its name would indicate, because after all, what does brachio refer to? Arm. So, whoops. It's going to go off to the arm. And what's cephalic refer to?
Brain.
Or cephalic really refers to head, but yeah, you got the right idea. Excellent. So this brachiocephalic trunk branches almost immediately to a blood vessel that is going to go to our arm. And what is the blood vessel that it branches off of to go to the arm?
Subclavian.
Say again?
The right subclavian, or?
Excellent. All right. Excellent. It is indeed the right. It'll get to the axillary, but that's not what it becomes at first. It is indeed the right subclavian. And this blood vessel that's going up towards the head,
The right common carotid.
now notice we had to make a point, and we should also be saying arteries after all of these as well to be specific in our description. Notice we've had to say right for both of those. Why didn't we have to say right for brachiocephalic trunk? Yeah, because there's only one of them. If it's not a paired blood vessel, we don't need to worry about right or left. But if it is a paired blood vessel, then we do need to say right or left. And as we mentioned, right subclavian eventually is going to lead to the right arm. Left common carotid goes to the left head.
Wait, was that a mistake?
I'm asking. There was a question. Does it go to the left head?
It goes to the right, right?
Oh, sorry, you're right.
Well, we only,
So it goes to the right head,
Right external and internal carotid artery.
which go where?
just
Uh,
to the head.
Well, to the right side of the head, right?
Right hemisphere.
To the right side of the head. Absolutely. We don't have a right or left head. We only have one head, but we have blood going to one side and then blood going to the other. Excellent. So what are the other two blood vessels? What's the middle one here? Excellent. Left common carotid. And where does that go?
Left side of the head.
Left side of the head. And the third blood vessel? Which goes to the left arm. Excellent. The good news is the pathways from basically this point on, in the right common, in the left common, in the right subclavian, in the left subclavian, they're symmetrical. Same on the left and the right. But when you're going to the right side of the head or the right arm, there's a little bit of extra tubing in the process. We have this brachiocephalic trunk, which we have to pass through first, whereas the left common and the left subclavian both come directly off the aortic arch. So if you think about it, the aortic, the blood vessels coming off the aortic arch go to 4 locations. Left arm, left side of the head, right side of the head. 4 distinct locations, but they do that with only 3 branches coming off of the arch. And that is because of the brachiocephalic. Any idea why? As I've talked about before, why is always one of those challenging questions to ask. But make a guess. I don't know if we're right or not, but we can at least make a guess. Why might we need a little bit of extra plumbing, a little bit extra tubing to get to the right side as opposed to getting to the left side of our head and our arms?
The heart's on the left side of the trunk.
Notice the heart's slightly offset. So maybe we need a little extra plumbing, a little extra tubing to get to the right side because the heart's offset a little bit. Who knows? We're speculating, but seems a reasonable explanation.
Longer distance. Yeah, maybe.
Exactly. Seems reasonable. Excellent. We've done a truly amazing job of drawing this on the board. And of course, if we look at the pretty pictures, we did all of that.
Professor?
Yeah.
Would another explanation also be due to the communicating arteries in the head? Because that allows for multi-directional. And so if you kind of let like the right side go a little bit, that's okay.
Sure. I mean, again, why is we're totally guessing, you know, like I said, I'm thinking of it from a plumber standpoint. If you have to go further, you need a little bit extra pipe, and that little bit extra pipe is going to have a name. Sure, it could affect things like pressure, and we're able to adjust for that in the cranial region. My concern then would be in the arms. If it caused a dramatic change in pressure that we had to worry about with those communicating arteries in the brain that they help to compensate for, then that would spell trouble for the right arm, and that's not usually the case. So, but it's reasonable.
Oh, I got you. Thank you.
Yeah. Again, I don't have a definitive answer. I'm just because, again, like I said, the engineer who designed all this did not leave us her footnotes, so we're speculating. So your guess is as good as mine. All right, here's the pretty picture, and again, the aortic arch, 3 blood vessels coming off of it, brachiocephalic trunk, left common carotid artery, and the left subclavian artery that allow us to go to those 4 locations. And the reason we're able to go to those 4 locations are because of the brachiocephalic trunk, which then branches immediately to enter into the right subclavian. Now, I'm just going to worry about the right side because, like I said, once you get to the subclavian, it is identical on the left and the right. As the names indicate, and again, like I said, all right, anatomists don't completely hate us. So notice the blood vessel that goes under the clavicle is the subclavian. The blood vessel that goes through the armpit is the axillary. The blood vessel that goes through the brachial region is the brachial. The one that lines up with the radius is the radial, with the ulna is the ulnar, and so on and so forth. So we have this basic pathway that gets us down to the hand, where we finally have a turnaround point with both the deep and the superficial palmar arches, which feed the fingers and get us down. And notice there's some other blood vessels, some alternate routes and things like that. Like I said, I'm not going to go through everything on this list, but this is our basic route to the side of the head. However, I mean, to the arm. However, there are 2 other blood vessels that I want to talk about. Do I have this here? Let's do that. Okay, let's cheat and do this here. We can do this here, but let's actually go back to my picture. And for convenience, I'll do this on the left side because I apparently left myself more room to draw there. For both, come on, get rid of that arrow. Thank you. For both the left and the right subclavian arteries, as we just finished mentioning, it goes under the clavicle, basically to the armpit, where it then feeds into the axillary artery. And again, this would be the left. I'm going to hold you to that standard. I should try to be good to it as well. But there are 2 other important blood vessels that come off the subclavian artery. One of those is one we already talked about earlier today, and that is that vertebral artery. Remember, this is the one that, as we mentioned, goes through the transverse foramen of the cervical vertebrae and ultimately feed into the brain. Forming part of that circle of Willis that we talked about. The other blood vessel, the one that arrow was pointing at in the picture, and we'll go back to that one in a second, coming off the inferior and anterior part of the subclavian is a blood vessel that is known as the internal thoracic. This internal thoracic helps to feed the blood vessels of the ribs, right, in those intercostal arteries. So if we cheat and go back to the pretty picture, Notice if we look closely at our image here, we can see how indeed, so yeah, the labels off. There is the common carotid going off this way, but this little blood vessel right here, that is that vertebral artery going up through the transverse foramen of the cervical vertebrae. And then notice here coming off the subclavian, going along the front of the thoracic cavity and feeding into those blood vessels between the ribs on the front is that internal thoracic. So while the subclavian primarily feeds the arms, notice it does have some major branches off of it that go to other places as well. We kind of already did this, so we'll go through it quickly. Not surprisingly, a common carotid branches into two blood vessels. One is the internal carotid and one is the external carotid. And as their names indicate, they tell you exactly where they go. The internal carotid goes into the skull, and as we saw, forms part of that circle of Willis. Whereas notice the external carotid is the one that primarily comes up to the skull and the skin, the superficial regions of the head. So their names tell you exactly where they go. The external carotid goes to the skull and superficial. Internal carotid goes inside to the brain. And notice, I think we've talked about this in this class. I'm pretty sure we talked about it in 430. Notice here where the common carotid branches into the internal and external is a structure that is known as the carotid sinus that contains structures known as the carotid bodies. These carotid bodies are actually, oh, I did that totally backwards. Are chemo and baro receptors. What does that mean? What does a chemoreceptor do?
It's a chemical receptor.
Yeah, it's measuring the chemical composition of the blood. What does a baroreceptor do?
Pressure?
Measures the pressure of the blood. Do you think it's useful to be aware of the pressure and the chemical composition of the blood going to your brain?
Yes.
Yeah, absolutely. This is one of the major locations where we are monitoring the condition of the blood so that we can make changes based on temperature, pH, glucose levels, sodium levels, all those kind of things that condition, and even pressure, where we can monitor the condition of that. And again, as I mentioned, we already did the circle of Willis, so I won't bother going through that again, other than to remind you that the starting point for this circle of Willis, again, are those two vertebral arteries coming off the subclavians and the two internal carotids coming off of the common carotid. And we did the rest of the circle of Willis, so we don't need to bother doing that again. All righty. And then, like I said, with the exception of that extra piece of tube in the brachiocephalic, the route to the left is identical to the route on the right. They are completely symmetrical. So like I said, if you're giving those driving directions, the only difference between the left and the right side, depending on where you're going, is if you're going to the left, you go straight from the arch into either the common carotid or the subclavian. Whereas if you're going to the right, you have to go to the brachiocephalic first, and then either into the common carotid or the subclavian. Only difference, which is nice. There are plenty of other asymmetries from the lefts and the rights, but here we don't have any. Well, we don't have many, just that one. All righty. As I mentioned, Yes, the aorta is sometimes referred to as the descending aorta as it drops down the backside. But like I said, our goal is to appreciate and understand the different regions of the body that are receiving blood. So it makes much more sense to me to use that big anatomical division, the diaphragm, to separate the descending aorta. The part above the diaphragm is, of course, the thoracic aorta, and the part below the diaphragm is the abdominal aorta. And we'll get to that in a second. What kind of things are in the thoracic cavity?
Heart,
Like the
lungs.
heart, lungs.
Heart and lungs are the big things. Doesn't the heart have its own circulation?
Yeah.
What about the lungs? Do you think the lungs have their own circulation?
Yes.
Yeah, didn't we learn about the pulmonary circulation? Notice, are there a lot of big structures in the thoracic cavity that need a large amount of blood that aren't already getting it?
Not really.
Not really, no. So notice when we look at the thoracic aorta, there really aren't a lot of major blood vessels coming off of it. In fact, really the only major blood vessels that are coming off of the thoracic aorta are these posterior intercostal arteries. Remember, the anterior get their blood from that anterior thoracic that's coming down, feeding into the anterior intercostals. So really the only major blood vessels coming off of the thoracic aorta are these blood vessels that sit in the costal groove of our ribs. And those blood vessels are the posterior and anterior intercostal arteries. So there's not a whole heck of a lot going on in the thoracic cavity. However, the same is absolutely not true for the abdominal cavity. For the abdominal cavity, there's a whole lot going on. So again, let's jump away from the picture and do a very simplistic drawing of this. Here we have our abdominal aorta coming down, and let's start simple. The termination, as I hinted at just before, when it terminates into not one, not two, but three major blood vessels that come off of the distal or inferior part of the abdominal aorta. Any idea what those three blood vessels may be?
The left and right common iliac, and then the medial sacral.
There you go. Perfect. We have the right and the left common iliac. And just like the common carotid, guess what the common iliac is going to branch into?
External and internal.
Yeah, into external and internals. Excellent. And what was the third one?
Medial sacral.
Excellent. The medial or median sacral is a blood vessel that primarily feeds the sacrum and the coccyx. So there are three main branches that come off of that. Excellent. So now we know the ending point. We also know the starting point. The starting point for our abdominal aorta is indeed going to be the diaphragm. So now that we have that, we can start working our way inferiorly and identifying the blood vessels of the abdominal aorta. Now, the first thing we have to figure out is if a blood vessel is paired or unpaired. So let's talk about the very first blood vessel. Just as we're coming down from the abdominal aorta, we hit our very first blood vessel. And is it a paired blood vessel or a unpaired blood vessel?
Unpaired.
Unpaired. Excellent. Just a single blood vessel. And what is that single blood vessel?
Celiac trunk.
Excellent. Ooh, there's that word trunk again. I know trunk means that this is a short, very short blood vessel that is going to branch almost immediately. And when it branches almost immediately, how many branches does it form?
3.
Excellent. What are they? Oop, hold on,
Common
something right-
hepatic artery.
All right, excellent. Common hepatic artery. Oops, down, down. Any idea where the common hepatic artery might be on its way to?
Liver.
Excellent, on its way to the liver. Spectacular. Splenic artery. Where is the splenic artery going to?
Spleen.
Excellent, going to the spleen. And the third one, hold on, it went off my chat window. There it is, left gastric artery. Guess where the left gastric artery goes?
Stomach.
Excellent. Guess which side of the stomach?
Left side.
Left side. Doesn't that mean the right side should get blood as well?
Does it innervate itself once it gets there?
Nope. Anyone know where the right
Half.
gastric artery comes from?
Don't they kind of connect?
Eventually, they will anastomose, but the right gastric artery actually comes off the common hepatic. So both the right gastric and left gastric are going to go to the stomach. The left gastric comes off the celiac trunk and the right gastric comes off the common hepatic. Excellent. First big blood vessel. What's the next one?
Is it the superior mesenteric artery?
Is that paired or unpaired?
Well, there's an inferior one, but that is that
Next to it? Yeah,
No,
you get-
no.
Excellent. We have another unpaired blood vessel that you have absolutely correctly identified as the superior mesenteric artery. Now you are correct. Way down here, down below it is also an unpaired inferior mesenteric artery. Any idea where these two go?
Intestine.
Yeah. Although we should probably be a little bit more specific, but absolutely you got the right idea. The superior mesenteric primarily goes to the small intestine, and the proximal large intestine. Whereas the inferior mesenteric primarily goes to the distal portion of the large intestine. Excellent. So you're right. It isn't truly paired, but not surprisingly, anytime you have a superior something, you have an inferior something. Every time you have a greater something, you have a lesser something. And so, yeah, that is an indication that way, but they're still considered unpaired blood vessels. You only have one. You do not have a left superior mesenteric and a right superior mesenteric. So it is an unpaired blood vessel. What's the next one as we work our way inferiorly?
Renal artery.
There we go. And I heard plural on that, arteries. So how many do we have?
Two.
2, absolutely. We have the right and left renal arteries. Excellent. And where do you think those go?
Kidneys.
Excellent. While you're sitting here calmly resting in your chair, something close to 25% of your blood is being directed to your kidneys to be cleaned and processed and made all good and happy. Excellent. Any more?
Gonadal arteries.
Excellent. Paired or unpaired?
Paired.
Excellent. We have those paired gonadal arteries. So a right and a left gonadal arteries. And where might those go?
Reproductive systems.
Yeah. Or specifically, what part of the reproductive system?
Testes and ovaries.
Yeah, collectively we refer to them as the gonads, absolutely. And so that means that they are different in men and women. Right? This is probably the very first meaningful difference we've talked about in men and women in anatomy and physiology. Obviously, in males, they're going to the testes, so they're also known as testicular arteries. And in the females, they're going to the ovaries, so they're known as ovarian arteries. Any other, actually, let's do it this way. Testicular arteries and ovarian arteries. Any other difference do you think between, besides the names?
Maybe the length and
Perfect, length. Whose are longer?
males.
Males,
Why?
because they're external. as
Yeah,
opposed to
absolutely. Right. The testes are actually located outside of the abdominal pelvic cavity. So absolutely, the testicular arteries have to be longer to get all the way out of the abdominal pelvic cavity, whereas the ovaries are nestled all snug as bugs and rugs inside of the abdominal pelvic cavity. So not surprisingly, they're shorter. Any other difference? I like that. Wider or size, we can think of this. Which ones do you think are larger or wider, bigger?
Probably male,
Why? Why would the males be larger?
because they would require more blood flow.
Why?
Constantly creating sperm.
Absolutely. Class started at noon today, right? That was 3.5 hours ago. How many gametes has a male made in the past 3.5 hours?
Thousands and thousands.
Yeah, probably millions and millions, absolutely.
A lot.
How many gametes has the female ovary made in the past 3.5 hours?
None.
None.
None. How many eggs has the female ovary made in the past 10 years?
None.
None.
None, right? In fact, female makes all of her eggs when she's in utero. When a female is born, she actually has all the gametes she's ever going to have for her life. She has to mature them, release them via ovulation. It's not that she doesn't have anything to do with them, but she's definitely not making any. And how many get ovulated during the course of a month?
Usually one.
Yeah, typically one, absolutely. So not surprisingly, this is a much smaller blood vessel there's less need because they're less metabolically active. Whereas obviously, like I said, males are making millions of sperm cells every day. I mean, quite frankly, we shouldn't be required to do anything else. We're so metabolically busy making gametes, we should just be allowed to sit and make gametes and not have to do any other work.
That's
But
the most man thing I've ever heard.
Exactly.
Deal. My anatomy teacher said.
There you go. Exactly. All the women should go to work and we should just be able to sit home. Excellent. So, all kidding aside, though, absolutely, there are some distinct anatomical differences. Yes, the maturation of gametes production and maturation of gametes in males is a more active process than the maturation of gametes in females. And so, not surprisingly, there is a need for a larger blood supply. So there are some differences. Again, first time I think really that we've talked about meaningful differences anatomically between males and females. And we talk about things like size and, lung capacity and stuff like that. But those, while statistically may be significant from a math standpoint, they're not truly important. Not that these are necessarily important as well, but they're certainly meaningful. All right. And then that brought us down to the inferior mesenteric. Did we forget any?
What about the
Lumbar artery?
So you are correct. Coming off the posterior side, and really the only ones coming off of the posterior side, there are some lumbar arteries that feed to the lumbar vertebrae. Absolutely. So those come, again, I know that's not a good job of showing them coming off the posterior side, but you get the idea. So,
excuse me, what about the phrenic arteries? Aren't they part of the abdominal aorta?
well, again, have I named all of the blood vessels that are here? Notice there's also adrenal arteries that come off of the abdominal aorta and things along those lines. But is the phrenic artery on your list?
No.
So then we don't care.
Okay.
Again, there are hundreds of named blood vessels in the body. Right, but absolutely, we are not responsible for all of them. You're just responsible for the ones on the list. So yeah, you're right. The phrenic, the adrenal, there are other ones as well, but we just need to worry about the ones that are on our list. So notice here, when we just look at the aorta, we can see there's our celiac trunk and the three branches coming off of that. Superior mesenteric, and then our renal, and then our gonadal, inferior mesenteric, two common iliacs, median sacral, and coming off the backside, those lumbar ones. Notice your book also has a nice picture that shows where some of these blood vessels are going. So notice, as we talked about, the splenic goes to the spleen. The left gastric goes to the left side of the stomach, whereas the right gastric comes off of the common hepatic, which, like we also talked about, goes to the liver. Here we see how the superior mesenteric is going to the small intestine and the proximal part of the large intestine, whereas the inferior mesenteric goes to the distal part of the large intestine. So we can see some of those additional pathways that we talked about of seeing where these blood vessels go. All right, questions on that? All right, that leaves us down to the arteries of the leg. As I mentioned, just like the common carotid branches into the internal and external carotid, our common iliac branches into the internal and external. The internal iliac, as our illustration shows here, basically feeds the organs of the pelvis, inside the true pelvis, those organs there, whereas the external iliac comes out to the leg, which feeds into the femoral. Notice we were talking before about the joints. Notice that popliteal artery is probably one of the classic examples of this arterial anastomosis. Basically, this whole elaborate mesh is kind of considered the popliteal artery, or part of the popliteal artery, giving all of those alternate routes so that no matter what position your leg is in, we can get the blood going where it needs to go. So again, down to the anterior tibial or the lateral, peroneal or fibrillar artery, down to the leg. Again, I don't think there are too many tricks down here, so I won't worry too much about that. Oh, that's why I have this. The one trick I did want to mention, it's not a trick, but your book isn't the best at noting. As you notice, when the external iliac branches into the femoral, it also branches into a blood vessel that basically goes deep along the bone of the femur. Your book actually refers to it as the deep artery of the leg. However, we're going to appropriately refer to it by its actual name, which is what?
The deep femoral artery.
Yeah, the deep femoral artery. Absolutely. So again, don't be confused by that on your list. It's there. Your book shows that it just has it slightly mislabeled. So again, we can clearly see it. We clearly know it's there. But other than that, I think there aren't any tricks to the arteries. All right. And with that, we have now distributed blood to every single part of our body. Question?
Yeah, I had a question. When you see like videos of people like standing straight up for long periods of time and then they like pass out or fall over or something like that, does it have to do with like one of these arteries or veins or something closing off or not getting the blood circulating?
It certainly wouldn't be the arteries. It wouldn't be an issue of the arteries that causes that. You're right. When people lock their legs, that it commonly happens. However, a lot of times I think it's also associated with stress. So I don't know if necessarily the locking of the legs is triggering the vasovagal reflex, or if it is somehow causing less blood to go back to the heart, so less volume could be pumped to the head. That doesn't seem like that would be it. My guess, and again, this is completely a guess. My guess is that there's something about that locked leg position which triggers a vasovagal reflex, basically an inappropriate activation of the vagus nerve, which causes systemic dilation of blood vessels. And that's what drops the blood pressure, and that's what causes people to pass out. So my guess is that it's still probably more neural as opposed to blood vessel related. Although, as we mentioned, if you do stand up too quickly, or if you think back to, when you were 5 and it was Friday night and you struck out getting alcohol that night. So instead, what did you and your buddies do? You'd curl up into a ball, right? You'd count to 20 and then you'd jump up real quickly to get dizzy so that you'd fall over and maybe even pass out as a result of it. I mean, as we talked about, hypotension can cause someone to pass out. I'm just not sure what the mechanism would be that would cause a dramatic enough change in blood flow that would cause that. So my guess is that it's probably neural, but I don't know that for certain. It's a great question, though. Any others? All right. Wow. Stunned silence. All right. I love those. Excellent. All right. So as I mentioned, we have now gotten blood to all parts of the body that we needed to get it to. So now we need to get the blood back. However, I've been talking for a while. Let's take a quick 10-minute break. Quick biology break. Use the restroom, grab something to eat, grab something to drink. Let's meet back here at 3:43, and we should be able to get through all the veins that we need to in that period of time. So just a quick 10-minute break, and we will restart. All right. Any questions? You guys are so quiet.
Nope.
All right. Excellent. See you guys in 10 minutes. Okay, I was gonna say I had to say something to get a reaction out of you guys. You guys aren't saying anything at all. It's very depressing. All right, just kidding. All right, let's go ahead and get started. All right, so we have gotten blood to all parts of our body. Let's focus on getting the blood back. Now, the good news is there is a fair amount of symmetry between the arteries and the veins. However, there are also some areas where it is very, very different. One of the big areas where it's very, very different is here in the cranial cavity, associated with the skull. Notice here, as you look at your list, there are a massive number of big, loose sinuses, which we know are big, loose, open veins. Why might something like that be important in the cranial cavity?
For material exchange.
Control pressure.
Right. So yeah, absolutely. We need an exchange of a lot of materials, but remember, it's going to be the capillaries that do that. But you've got the right idea. Pressure is a major issue. If I get a little bit of swelling in my hands, some extra edema in there, right? It makes my hands a little bit tighter when I try to grip. It's a little harder to get my wedding ring off when I get to the bar for Thursday night, because this is my Friday and it's time to party, right? But it's not that inconvenient. However, if I start getting edema inside of my cranial cavity, could that cause problems?
Yes.
Absolutely. And so we need to make it as easy as possible to drain the fluid out. The other thing that hopefully you remember from 430 is also surrounding our brain and inside of those ventricles is our cerebral spinal fluid. Remember, it is produced coming out of our choroid plexus, which are basically capillaries with those ependymal cells associated with them. And then we want to be able to then reabsorb it. And so one of the main places it gets reabsorbed back into is into those big dural sinuses. If you remember, our dura mater is a big, huge, thick, two-layered fibrous connective tissue structure that has some folds that come off, like the falx cerebri and falx cerebelli, to help to stabilize the brain and hold it in place. And when those layers separate, there's some big sinuses in there. And so, of course, one of the biggest ones is our superior sagittal sinus, a big, large sinus that is located in the falx cerebri, between the two layers, basically sits along the top of the longitudinal fissure between the two hemispheres of our brain. And as we mentioned, anytime we have a superior something, what are we going to have to go along with that? Inferior something. And sure enough, there along the midline, also in the falx cerebri, is a much smaller midline sinus known as the inferior sagittal sinus. Not only is the inferior smaller, but it also doesn't go all the way to the posterior part of the cranial cavity. So you will notice there is an additional shortish but very straight sinus that carries the blood to the posterior part of the cranial cavity. And what do you think we call that short, straight sinus?
The straight sinus.
Straight sinus, absolutely. There is also a big, large, dare I say, cavernous sinus located just posterior to the maxilla, kind of by the orbit of the eye, known as the cavernous sinus. And notice all of these sinuses, what color? Change colors. The superior sagittal, the straight sinus, the cavernous sinus, all of these feed into this midline sinus that is S-shaped. And because it is S-shaped, it gets the name the sigmoid sinus. And that sigmoid sinus takes all of this blood and feeds it into our very large internal jugular. Notice, just like we have an internal carotid and an external carotid, we have an internal jugular and an external jugular. The external basically drains blood from the skull and from the superficial part of the head, and the internal jugular from the cranial cavity. And not surprisingly, the internal jugular is way, way bigger because, again, we want to make it as easy as possible to get that blood out of there. But I do want you to notice one other thing. I'm going to go ahead and erase the pictures that I've drawn so far. It's a little tricky from this picture that we see here. It doesn't do the best job of showing it. So actually, let's cheat and draw this. If we were to look at the cranial cavity from the top, as we mentioned, the superior, inferior, straight, cavernous, sigmoid, all of those are basically on the midline. Now, the sigmoid does branch to feed into the two internal carotids. And we have two internal carotids that come off the two sides. But notice there isn't really much of anything to drain the lateral aspect of the cranial cavity. Wouldn't it be convenient to have a blood vessel that kind of ran along the inner surface on the lateral aspects to help to drain blood into that sigmoid sinus? What might something like that be?
Would be the transverse sinuses.
Yeah, so these lateral ones, and again, this picture doesn't do the best job of showing it, but there to the lateral aspects are these transverse, a right and a left transverse sinus that also helps to collect and drain that blood primarily into the sigmoid. Almost all of these feed into the sigmoid. And again, these are veins, so there's lots of collateral routes, but the primary route is to feed these things into the sigmoid colon, which feed them into the internal jugular, with that very important goal of getting the blood out of the cranial cavity. All right. Questions on that?
Yeah, Professor, one question. Going back to that question I asked earlier, how do the arteries like the anterior cerebral, middle cerebral, and posterior cerebral connect with these sinuses?
Through capillaries. Remember, the turnaround point for all of these things are going to be capillaries. So within the nervous tissue, you have all of these tiny collateral blood vessels that are feeding off into the different regions of the brain, and they are feeding blood into capillaries that are surrounded by astrocytes to form that blood-brain barrier, exchanging materials with the nervous tissue, and then feeding into venules that feed into these.
Right, but also like, would the middle cerebral, so like if we're trying to trace a drop of blood, we would get to the middle cerebral.
Oh, I see what you're saying. So I would say, what I would recommend is to. It's not quite as simple as that, especially because we have the arachnoid fluid. There are a tremendous number of smaller blood vessels in the nervous tissue that we have not been responsible for identifying.
Mm-hmm.
So you've got the right idea. For instance, stuff that are going into the anterior cerebral are more likely to drain into the cavernous sinus eventually, for instance. Things that are on the lateral, like the parietal or the temporal, lobes are going to be more likely to feed into the cavernous, the transverse along the sides. So again, there are going to be many routes that these things are going to take, and I don't think you have to worry about that level of precision in the nervous tissue, because again, the vascularization of the brain is easily more complex than it is anywhere else in the body.
Got it. Okay, so if you ask us a question on the exam of like going from a specific spot all the way back to the heart, it wouldn't be from the heart, from the head.
One of your questions was to the head, but notice it was the superficial head, right? So notice if you go to the superficial, temporal artery, that's most likely going to feed into the temporal vein, which is going to feed into the external jugular, and so on and so forth.
Right, that's simple enough. It was just the sinuses that I was getting confused on.
Yeah, so you're right. How they directly connect is indeed confusing and more than we have to worry about in this class.
Got it. Thank you.
Now, aside from being named different, internal external carotids versus the internal and external jugular, notice there is no common jugular. Instead, as you can see, external jugular feeds directly into the subclavian vein. And notice the internal jugular and the subclavian come together. And when they fuse and come together, they form a brand new blood vessel. And what is that brand new blood vessel that is formed by the internal jugular and the subclavian coming together? If you're not for sure, you can cheat and read it off the screen.
Brachiocephalic vein.
Right. Notice this is blood coming back from the head, coming back from the arm. And notice it doesn't just say brachiocephalic, it says right brachiocephalic. What might that indicate?
There's a left.
There's a left, exactly. Whereas we only have one brachiocephalic artery, we have two brachiocephalic veins. So in the case of the veins, the pathways for the left side and the right side are completely symmetrical. And those two, the right and left brachiocephalic veins, come together to form what singular blood vessel?
Superior vena cava.
Superior vena cava. There you go. Perfect. So whereas we have to worry about that extra bit of tubing to the right for the arteries, there's no extra tubing for the right versus the left for the veins. They are completely symmetrical. Same thing for the arms as well. Completely symmetrical. And the main pathways are indeed similar, identical to what we saw in the arteries. We have radial and ulnar veins that feed into the brachial vein, that feed into the axial, the subclavian, and then so on and so forth. But as we can see, there are more anastomoses and some additional blood vessels here as well. There are two I want to point out to you. Hold on. I guess I stuck with this here. All right, so I show the main pathway, but what isn't labeled on this, but I want to show you, is there is two additional blood vessels that I want to point out. Whereas the brachial vein is deep and kind of runs along the bone, there is a vein that is on the lateral aspect of the arm, and there is a vein on the medial aspect of the arm. Anyone know what this blood vessel on the medial aspect of the arm might be called? So I only have a list of the blood vessels I was responsible for. I might be able to tease it out.
Median cubital vein.
A median cubital is not a bad guess, although the median cubital, as its name would indicate, basically goes through the midline part here of the elbow.
Is that the basilic?
It is indeed the basilic vein, absolutely. Notice not basilar. Basilar was the one that we saw in the brain. Basilic vein, excellent. Basilic kind of means base. What the heck's the other one called?
Cephalic vein?
What did we say cephalic meant again?
Head.
Head. Why the heck do we have a vein in our arm that's called the head vein? Well, I'll answer that for you. It turns out, from a developmental standpoint, when we're little embryos growing and our arms are just little buds of tissue that are sticking out, our arms tend to be in an upward position. So this blood vessel right here on the lateral aspect of my arm is right next to my head, whereas the other part is the base away from it. So they are the basilic and the cephalic veins, named from that orientation during development. So like I said, I think most of the pathway is kind of simple. These two, I think, because of their names, are a little unique. So I wanted to point that out.
Are those common veins for like a lab draw, just based on their
No, the
location?
median cubital is usually the most common for a lab draw. Although, like you said, they are superficial, so they are easier to get to than the brachial, for instance. Yeah. Notice, not surprisingly, like the thoracic arteries, there's not a whole lot going on in the thoracic cavity, although a little bit more. You'll notice there is a little bit more going on in the thoracic cavity. And again, it's a little bit wonky. So that's when I want to spend a little bit of time talking about it. As you can see, we have all of these intercostal veins. And the intercostal veins on the right side of the body feed into this long blood vessel known as the azygous vein. Notice the azygous vein feeds into the superior vena cava before it reaches the heart. And it pretty much drains the right side of the thoracic cavity. So, of course, we have a right azygous and a left azygous, and the left azygous does the same thing on the left.
No.
No, that'd be way too easy. Instead, we have a blood vessel that tries to do the same thing on the left. It helps to collect blood from the intercostal veins and is bringing its way up, but it gets tired. And so it only goes halfway. And because it only goes halfway, it is called the hemiazygous. So the hemiazygous drains the inferior left side. But don't we still need to drain the superior left side?
Yep.
Yeah. Well, lucky for us, the hemiazygous has help. And that help comes in the form of the accessory hemiazygous. So the accessory hemiazygous drains the superior left, hemiazygous, the inferior left, and both of them feed into the azygous. So the azygous drains the right side and collects all the blood to deposit it into the superior vena cava. And don't ask me why, I have no idea, just the way it is. Weird, interesting. We need to learn 3 names. We really should have to learn just 2, a left and a right of something, but it is what it is. Interesting, but not as bad as the head, and also not as bad as the abdominal pelvic cavity. Let's take a look at this and let's draw it again. Let's keep this picture, so let's add a second one. Obviously, the big blood vessel that is going to drain blood from the abdominal pelvic cavity is the inferior vena cava. And of course, it has to pass through the diaphragm. So we can go ahead and draw our diaphragm again as well. And we know it's going to continue up beyond that to the heart, feed into the right atrium. And once again, starting superiorly, we can start to identify some of the blood vessels that feed into the inferior vena cava. What's the first set of blood vessels that feed into the inferior vena cava?
Hepatic vein.
Hepatic veins. There are multiple hepatic veins, anywhere from 2 to, I don't know, maybe in some cases as many as 6. But regardless of how many there are, where do they come from?
The liver.
Hepatic portal vein.
Liver. Yeah, they come from the liver. Absolutely. Excellent. What's next?
Superior mesenteric.
Is it?
Is it the hepatic portal?
Hepatic portal.
Nope, I don't see a hepatic portal. In fact, if I follow it down, next two I see are the renal veins. Guess where the renal veins come from?
Kidneys.
Excellent. What should be under the kidneys? What were under the renal arteries?
Gonadal.
Excellent. And notice, sure enough, if we look at our picture, lo and behold, there's our right gonadal. And wait a second. Where's the left?
Off of the left renal.
There you go. Absolutely. We do indeed have to worry about our gonadals, not worry about, but we need to drain the blood back from our gonads in both males and females. But for some odd reason, and again, absolutely no idea why, the right gonadal. See, now every time I stumble with my words like that, you guys really are going to think that I've had an edible. The right gonadal feeds directly into the inferior vena cava, whereas, and again, I have absolutely no idea why it would be organized this way, but the left gonadal feeds into the left renal. A very, very weird asymmetry that I can't even begin to hazard a guess as to why you would set it up this way. It just is. But it's consistent. Males and females, this is the way it's set up. And notice if we continue down from that, then really all that's left is the terminal, or really the starting point. This would be the starting point where we have our right and left iliac that come together along with the median sacral to form the inferior vena cava. Notice there's a whole lot less going on here in the abdominal aorta. And you kind of hit on why. Notice there is no mesenteric. Superior or inferior. There's no gastric. All right, all of that stuff. What do all of those things have in common? What are they associated with?
So the parasympathetic.
Well, not a bad guess, although I think Samuel's closer. The digestive system, like the stomach, like the small intestines. Notice if you think of it from an artery standpoint, from an artery standpoint, we have a left and a right, a gastric. We have a superior and an inferior, a mesenteric. We have the common, a hepatic artery. We have all these things that go to the stomach, to the small and large intestine, and to the liver. And of course, why do they go to all of these places?
To supply blood.
To supply the blood to these areas to do what? Yeah, to absorb, break down, and absorb the nutrients. From the food we ingest. Now, once you break down all of those foods and get all of those nutrients, do you necessarily want to take them and dump them directly into your cardiovascular system for your heart to distribute throughout the body?
No.
I know this may be a hard question for you guys to answer because I know every single person in this classroom eats a completely well-balanced meal with just the right amount of micro and macro nutrients that you need in the food that you ingest to maintain perfect balance within your body. But some people might have a donut and Diet Coke for breakfast. And are they necessarily getting everything they need from that?
No.
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 So after this, we have blood that is nutrient rich, but not necessarily nutrient appropriate. So before we can dump this back into the cardiovascular system, we have to send this blood someplace. And where do we send this blood?
The kidneys.
Kidneys is a good place to process it, but that's really about getting waste. There you go. Amy's got it. Where we're going to send this is actually to our liver. And our liver is where magic happens. And after that magic, our blood goes from nutrient rich to now the blood is nutrient appropriate. So we need that processing by the liver. Oops, there. And the way it gets there, all of these structures feed into veins. The gastric arteries feed into gastric veins. The mesenteric arteries feed into mesenteric veins, but all of them feed into the hepatic portal vein. And this hepatic portal vein brings the blood to the liver for that magic to happen. And again, we get to the digestive system, we'll talk about what the magic is. But once we're done with that magic, once it's nutritionally appropriate, then it leaves the liver into those hepatic arteries. And that, of course, as we saw, feeds into the inferior vena cava. Again, details matter in this class. That portal is a big difference. Oh, gosh. Hepatic vein. Big difference between the hepatic portal vein and the hepatic vein. Hepatic portal vein is bringing the nutrient-rich blood from our digestive system to the liver, where it gets processed. and then our hepatic vein takes the blood from the liver and feeds it into the inferior vena cava, which means it goes to the heart, and from there it goes everywhere. So notice there aren't nearly as many blood vessels feeding into the inferior vena cava as there were coming out of the abdominal aorta. And I believe, here we go. Here we see that hepatic portal system. Notice here, feeding into the liver is that hepatic portal vein. Notice receiving blood from the stomach, receiving blood from the large intestine and the small intestine, both parts. All of these things are feeding the blood that is nutrient rich into the liver to be processed. And then once in the liver, it is released via those hepatic veins into the inferior vena cava. So digestive blood has to go to the liver first for processing. Lastly, as we mentioned, the appendages are pretty simple and straightforward. Really, the only big difference in the leg and the arms, as we mentioned, is more blood vessels. And one of the big blood vessels, and I literally mean big blood vessel in this form, is the great saphenous vein. As you see, the great saphenous vein basically starts all the way from the posterior part and inferior part of the foot, comes all the way up the medial aspect of the leg, and basically feeds into the femoral artery right before it feeds into the external iliac. It's the express lane, basically from the bottom of the foot all the way up to the upper leg. It is the longest vein in the body. And originally, if you were going to have like bypass surgery in the heart or require some other type of vascular transplant, this would be the blood vessel they would most commonly take. They would harvest your great saphenous vein and use that to form the connections necessary to bypass blockages of the heart. Because they figured it came from you, there was no chance of it being rejected. And so, you know, and again, it's a collateral pathway. There's lots of pathways back from the legs. So yeah, it makes it a little harder for your circulation of the legs, but it's more important that the heart gets the blood that it needs. However, more and more they're finding that these are rarely rejected. So most of the time now, those blood vessels are coming from cadavers or other sources as well. So typically they're not harvesting it from you anymore, but in some instances they still will. And of course, anytime there's a great something, what else is there? A small something. And sure enough, the small saphenous vein also starts at the foot, but basically goes up to the popliteal vein, which also has an elaborate anastomosis and feeds into it. And here you see on the anterior side how it's coming in to that popliteal vein on its way back. So again, most of the pathways are similar, but there are a couple extra blood vessels that you need to know here as well. All righty, questions on that? Like I said, I appreciate we did not go over every blood vessel on that list, but that was never the goal. I wanted to talk about some of the basic pathways that you are responsible for. And as you saw, there were some areas that were a little bit more unique, had some asymmetries, had some unique characteristics or challenging characteristics. And I wanted to make sure that we covered those at least superficially. I don't expect you to have mastered them by now, but hopefully me exposing you to them will help you as you're studying this moving forward from here. All right. Any questions on any of that? All right, excellent. Again, you have 5 days now till we meet again. So hopefully that is plenty of opportunity for you to continue to work on this material, challenge yourself with material, become comfortable with this material as we move forward. All right, because remember, you only have 2 more weekends before your first exam. So this is practically half of your study time. So definitely take advantage of this, and I will see you guys on Tuesday. All right. Have a good day.
Thank you.
Thank you.
Yep.
Thank
Bye.
you.
Thank you.